Hi everyone. Hi everyone. I hope I'm perfectly audible to everybody. I hope I'm perfectly audible to everybody. Okay, so welcome to Unacademy NEET English. I'm your biology educator Ambika Sharma. So today we are going to complete the chapter excretory product and its elimination. We are already done with the part 1 in which we discussed about the excretion, osmoregulation, what is the meaning of homeostasis. Then we talked about the different types of excretory waste, different types of organism, what sort of excretory organs do they have and what sort of waste do they have. So this is what we have already covered in the last class. So in today's class, we will start our class from the structure of nephron. So if you will check our channel, that is Unacademy NEET English, you will find that I have uploaded the part 2 of the anatomy in which the tissue part is there. So tissue part, that basically that is not given in the new NCERT, but it is still the part of your syllabus. And tomorrow I will upload the part 3 in which we will complete that chapter, okay, as promised. And after that, human health and disease again in the recorded format you people are going to get. So today let's finish this particular chapter, okay, that is our target for today. We will finish this particular chapter, but let's have a quick revision. So in the last class, I told you about the defecation. Yes, everybody, I told you about the defecation. Then we discussed the excretion, the difference in these two. These basic definition like what is excretion, osmoregulation, osmoregulators, conformers, then different types of nitrogenous waste. We discussed the energy requirement, dual excretion, right? So different phylums, their organism, their excretory waste, their excretory organ. So that's what we covered in the last class along with the NCERT highlight, okay, along with the NCERT highlight. So here you guys can see the internal structure of the kidney as well, right? You know that in the case of kidneys, that is what we discussed, that firstly all the nephrons, they are together with the help of connective tissue, right? That is the white fibrous capsule will form. The fibrous tissue is basically will be formed. Then there is the adipose capsule. So here you are talking about the fat, right? Fat layers. And then you are going to talk about the renal fascia. Is that clear? So that will help to attach these kidneys to the abdominal wall. So that is what we covered in the last class, right? Along after that, see this. Kidneys, retroperitoneal, dark red, slightly flat, all the points and the notes we made in the last class. So I hope you are done with this particular portion. So today we will complete this chapter and then I will upload this particular PDF in our official Telegram group. Okay, right? So let's start the class today, and as I said, what are we going to talk about? We are going to talk about the nephron first. Okay, what are we going to discuss first? We are going to talk about the nephron first. So let's have a look. Everybody please make the diagram. See this. This is what the nephron is. So we know that it is the structural and the functional unit of the kidney, and each kidney is having around 1 million nephrons. Is that clear? So you'll find 1 to 2 million here, 1 to 1.5 million. So as per NCERT, we will go here. So it is around 1 million nephrons are present in each kidney. That is what we discussed. Okay. So now obviously if these kidneys are there, then the blood supply will also be there, na? Right? The oxygenated blood will come, the deoxygenated blood will go. So few things, I'll start here and then we'll discuss that. Just let's suppose you have the renal artery, right? What do we have? We have the renal artery, right? That will carry the oxygenated blood to the kidney. Renal word attached, basically it is related to the kidney. Now obviously this artery is having the oxygenated blood and it has to pass the blood to the all the nephrons present in the kidney. Then this artery will further break, it will form arterioles. Okay, it will form arterioles, it will form peritubular capillary networks. So blood supply is also there, because ultimately how the urine is going to form? Urine is just the filtration of the blood. How the urine is going to form? Urine is just the filtration of the blood. Is that clear? So when you look at this particular structure, so when you see this cup-shaped structure, okay, what do you call it? You call it as woman's capsule. Okay, you call it as woman's capsule. This folded part, for this folded part, the terminology is convolutions. So it is your proximal convoluted tubule. It is what? It is proximal convoluted tubules, and the short form here is PCT. Then you will see this U-shaped structure which is known as Henle's loop, and you can also call it as loop of Henle. Okay, and then this part is what? This is the descending limb. So this is the thick, because it is descending like this. This is the thin part. So this is thick and thin descending limb. What's that? It is thick and thin descending limb, right? Right. So this is like this. This is the thick, and this one is the thin ascending limb there. What are they, people? They are the ascending limb. So that's what you have to remember. And this one is the DCT, that is distal convoluted tubule. Am I clear? So what is it? It is the DCT, that is distal convoluted tubule. Please keep it in your mind. The structure needs to be very clear, because today we will see what type of cells are making these parts. Okay, and then we'll talk about their functions. So this is your collecting duct. So DCTs, they are having this collecting tubule. All of it will open in this collecting duct. So many collecting duct will together form the duct of Bellini, P-E-L-L-I-N-I. Okay, so that's what we have. Now, when I talk about the blood supply here, so in that case, you'll see that this cup-shaped structure will have the tuft of capillaries. What will it contain? It will contain the tuft of capillaries and all that blood vessel, right? See this? They are covering this part also, and even we'll see a U-shaped structure, right? We will also see a U-shaped structure around this particular loop of Henle, like U-shaped capillary network also around this loop of Henle. You people are going to see. Is that clear? Yes, everyone. Is that clear? Done. So it is like that. And here also you'll see, look at this, the entire capillary network is present. Okay, that's how it is. So basically blood vessels are going to cover it. Okay, so this portion, when you talk about this tuft of capillaries, this part here, the one which is going there, right? This one, it is the afferent arteriole. What's that? It is the afferent arteriole. Am I clear? This tuft of capillaries, what is it? It is glomerulus. Firstly, understand the structure. It is glomerulus. This one which is leaving this part is efferent arteriole. What do you call it? We call it as efferent arteriole. Are you getting my point? Yes, students. What do we call it? We call it as efferent arteriole. Now, this is actually the peritubular capillary network. Peritubular capillary network. Then look at this U-shaped structure. It is actually known as a U-shaped network of capillary. It is known as vasa recta. Okay, beta, it is known as vasa recta. So this one is the ascending limb of vasa recta. This one is descending. I'll discuss this portion separately in the countercurrent mechanism. So you please pay attention here. Okay. Now, this is the peritubular capillary network. So this is the structure of the nephron. So each and every part clear. So remember this glomerulus and woman's capsule. Together we call it as Malpighian capsule or renal capsule. You can call it as Malpighian capsule, or you can also call it as renal capsule. So that's what you have to remember. These two together, glomerulus plus Bowman's capsule. Am I clear here? When it comes to the diagram part, everything is clear? Everything is clear? Yes, please. Everything is clear? Yes, please. Done? Yes, please. Okay. Any doubts here? So it is the Malpighian capsule or Malpighian body. Okay. Any doubts? Any doubts? Structure clear? Okay, so here you people can see this particular diagram as well. Okay, so now here I am marking it. So I told you this is loop of Henle. This U-shaped structure is loop of Henle, and this one is vasa recta. So if it is the, see, this is the descending limb of loop of Henle, and this one is the ascending limb of vasa recta. And here you have ascending limb of loop of Henle. So this is your descending limb of vasa recta. That's what you have to keep in your mind, and I'll tell you why. Okay, then I'll tell you later why is it so. Quick, right? Then I will tell you later that why is it so. Guys, quick, answer it to me. Thank you so much, Anis. Done? So this part is clear? Are you sure? This part is clear? Are you sure? So diagram clear. So let's talk about its structure, and we will talk about the woman's capsule first. So you make sure you please make the notes as well, okay? So let's talk about what? Woman's capsule. So this cup-shaped structure that we have discussed. So before writing the notes, let me tell you, right, before writing the notes, let me tell you, just see, this is that cup-shaped structure, right? I'll give you the overview here first. So when you look at this outer, right, this is the outer wall of this woman's capsule. So in this outer wall, you will not find any specialized epithelial tissue here. You will find the simple epithelial tissue. But here you will see the specialized epithelial tissue is there. Here we will see that some cells are modified. We'll talk about it. And when you talk about this tuft of capillary, this is the blood capillary, and we know that here also you are going to see blood capillary. You know that they are mainly meant for what? For filtration, for diffusion. So here also you will see that there will be the cells, endothelial cells are there, and in between we are going to see some pore-like structures as well, right? So, and when blood will pass to this area, here we know that the filtration will occur. So this is the complete structure, and this is the structure in which our first step of urine formation, that is glomerular filtration, will occur. Are you getting my point? Here it will happen. So as I said, in the woman's capsule, the outer wall is having the simple squamous epithelial tissue, but here it is the specialized squamous epithelial tissue. So if you know what is the meaning of squamous epithelial tissue, then good. If not, you know that we have discussed it in the structural organization in animal. This is important. So squamous epithelial tissue, it is having the irregularly shaped cells, right? What does it contain? It contains irregularly shaped cells, right? So, right, irregularly shaped cells. So it is also known as pavement epithelium or tessellated, right? It is also known as pavement epithelium or tessellated epithelium. Is that clear? Pavement epithelium, and you can also call it as tessellated epithelium. Okay, students, so that is what you have to keep in your mind. So even you have you have this blood capillary, very thin they will be having the endothelial cells here, right? And whenever you are talking about the epithelial tissue, we know that it lies on the basement membrane. And here in this inner, right, inner Bowman's capsule, here also you will see that there are some, some specialized cell, right, that are known as podocytes. I'll show you the pictures as well. So poda mane feet. So here you will see these type of projections. So they are placed in such an intricate manner, they are going to form the slit pores. And even in endothelial cells also, you will see the fenestrae, the gaps there, and you will also find the basement membrane in these two, because epithelial tissue, it lies on what? It lies on the basement membrane. So this is in general about the Bowman's capsule. So it's a large double-walled cup-like structure. Is that clear? Is that clear? So it's a large double-walled, it's a large double-walled cup-like structure at the beginning of nephron. It is at that proximal part, of course. So it is having, it lies in, now you know these terms. So it lies in the cortex of kidney. Where do you see this? It lies in the cortex part. It is not in the medulla part. Yesterday also we talked about it. It is in the cortex part. Is that clear? It is in the cortex part. So it is having, yes. Okay. Yes. So whatever we have just studied, we have to write that only. So it contains tuft of capillaries called glomerulus. Inner wall of Bowman's capsule, also called visceral wall, is in close association with glomerulus. Okay, so this one is in close association with glomerulus. Understood? And here you will see that the outer wall, okay, so outer wall of Bowman's capsule is in continuation with the rest of the nephron. So as I told you earlier, and I'm writing it here, so glomerulus plus Bowman's capsule together they form renal capsule or Malpighian capsule or Malpighian body. So this is what we call it, and this part is important. Please write down quick. Please write down quick. Yes, please. Done, beta? Understood? Understood? So this part is what? This is actually the lumen, right? This is what? This is the lumen part. So now this, so the glomerulus, which is a blood capillary, is having endothelial cells, is having what? It is having endothelial cells, right, that lie on the basement membrane, told you already. that lie on what? Beta, this information is important even for the urine formation. You should know all that things. So please pay attention. So that lie on the basement membrane. So these endothelial cells, if I have to show you the diagram here, right? So these are the endothelial cells, and see, this is going to be the basement membrane on which it will lie. So these are that endothelial cells of that blood capillary, okay, and the gaps here, these are known as fenestrae. So it is not continuous, they are having the gap in between. This is the fenestrae here. What is it? It is the fenestrae, the gap in between, and this is the basement membrane. So yes, you can say that it is the basement membrane, that is a right, it is that basement membrane that is a continuous layer present between blood in capillary and the inner wall of Bowman's capsule. Is that clear? So that is how it is present. Please write down. Okay, please write down. Quick. So these gaps are fenestrae. Okay. Done, beta? And I would say here, the instead of inner wall and the lumen of Bowman's capsule, I'll tell you how. Because here in the, because this inner wall of Bowman capsules have, inner wall of Bowman capsule contain modified epithelial cells called podocytes. This is very important information. So what are they, students? I just told you they are the podocytes, right? They are the podocytes, okay? So podocytes are arranged in such an intricate manner that they form slit pores, okay, that they form slit pores. So told you already this thing, right? Slit pores. So imagine if this is the endothelial cells, right, and here you will see that, you know, podocytes are present and they are arranged, poda mane feet, so they are arranged in such a way that in between you see the slit pores. So that is why on the basis of that we are saying that it is the basement membrane that is the only continuous layer in present in between blood in the capillary and the lumen of Bowman's capsule. So if you see this particular diagram, people, please have a look. So here in the glomerulus also told you already that the cells will have the space in between. These are the fenestrae, right? Fenestrae, obviously it lies on the basement membrane, and here you have podocytes which are arranged in such an intricate manner that they will also, just say, imagine, let's suppose this is one cell having feet-like appendages, this is another cell. So if it is fixed like this, if it is placed like this, right? So if you will see what? You will see some slit pores there. You will see some slit pores there. So that is what I'm trying to tell you. That is why I'm saying that it is the endothelium, it is the basement membrane. Right, it is the basement membrane that is actually the continuous layer present here in between two. Is that clear? Done? Done, beta? Okay. Sure? Right? Right? Any doubts? So here you can see, you can even mark it here only, that the outer wall of woman's capsule contain unspecialized, like they are not modified, they are the normal, usual, unspecialized squamous epithelium, right? Squamous epithelium, that is it. So I hope this part is clear to everybody, okay? I hope this part is clear to everybody. So this is what you have to remember. So this is the area, right? This is the area, this is the part where the first step of urine formation, that is ultrafiltration, will occur, okay? This is the first step of urine formation, that is ultrafiltration will occur. Am I clear? Am I clear? So glomerular, that is the blood capillary, very thin they are, we know it very well, right? So because they are very thin, that is why, my dear students, the filtration will occur here, and here in this part, the filtration of blood will occur, and that will form the filtrate, which will finally, right? Which will finally form what? Which will finally form the urine. Am I clear? So any doubt here? Yes, please. Any doubt here? Done? Okay. So let's talk about its next part. So yes, so let's talk about the next part here. So next is the PCT. Okay. Next structure that you have to discuss here is PCT. Am I clear? Sure? Sure? So you can just write down that woman capsule is followed by a small neck like structure which is lined by, only a very small neck like structure which is lined by ciliated epithelium. So here you will see ciliated epithelium. Cilia, you know that, short, you know, some hair-like appendages. So this ciliated epithelium will be present here, right? So if I have to show you the diagram, so I will say, let's suppose this, and here a very short neck-like structure. Here you will see the ciliated epithelium, and then, right, and then you will see PCT, proximal convoluted tubule. So when you talk about the proximal convoluted tubule, here you will see the epithelium is going to be brush bordered. That's a PYQ again, I'll explain it. So it is the brush bordered cuboidal, simple cuboidal epithelium. Why am I writing the word simple? So simple means it is going to be single layered. So brush bordered simple cuboidal epithelium. What's that? Simple cuboidal epithelium. But why do we say brush bordered? See, as I said, here it is the neck region where you will see the ciliated epithelium. This is a very short region, and then comes the PCT. Now, see, here the blood will pass, right? From here, the blood will get filtered, okay? Blood will get filtered. We will discuss that in urine formation. That filtrate will come here in the lumen. Actually, this filtrate is not having everything which is unimportant, which we need to throw out from our body. No, we have to reabsorb many things from this filtrate. So now when you look at the PCT, which is having the brush bordered epithelium. So brush bordered means it will have small, first of all, cuboidal epithelium means cube-like cells. Right, cube-like cells will be there. Now when I'm saying brush bordered, so here you will see that at their free ends, you are going to find the microvilli. What will you see here? You are going to find the microvilli here, which are small finger-like, right? Small finger-like structures, right? Small finger-like structures that increases the area for absorption. Am I clear? That increases the area for absorption. Are you getting my point? Like in our small intestine, we have the villi. Now these cells are also heavy. So in small intestine, we have the villi. We have that finger-like projection in our small intestine. So here cells are having that small finger-like projection, which we are calling as what? Which we are calling as microvilli. Am I clear? So these microvilli, they are going to increase the area for absorption. So here, right, this epithelium looks like this. So we call it as brush-bordered epithelium. Why? Because 70 to 90% of the filtrate, right? So we will say maximum reabsorption of filtrate occur here. Am I clear? Maximum reabsorption of filtrate occur here. So this is what you have to remember. Write down. Quick. Done? ओके बेटा, चलिए. सो ओके, so next is the loop of Henle. Understood? So PCT, maximum reabsorption of filtrate occurs here. So can we say that it will pass the filtrate from this lumen part to within the cell, right? And brush bordered, and what I just said, simple cuboidal brush bordered. So this is a previous year question, beta. This is a PYQ, that PCT is lined with simple cuboidal brush bordered epithelium, right? So if you know that tissue part from structural organization in animal, then you will understand it very easily, okay? Right? So this is what we have here. So maximum reabsorption occurs here. And here I told you this neck region will have what? This will have the ciliated columnar. Again, ciliated, not columnar, ciliated cuboidal epithelium. So here also cells are cube-like, but they will have cilia instead of microvilli, right? Like this, like this. This sort of epithelium will be there. Am I clear? Right? So next to it is what? Next to it is the loop of Henle. Okay? So it is, so it's loop of Henle. So it is present, it's a U-shaped structure present in between. It's a U-shaped structure which is present in between PCT and DCT. Okay, PCT and DCT. Here you will see thick descending limb, right? And this thick descending limb is lined by, yes, this is the descending limb, this is the ascending limb. And we know it already that they'll be present in the medulla region, right? Where will they be present? They'll be present in the medulla region. Medulla region of what? Medulla region of your this kidney, right? So here in the surrounding, we know that vasa recta is there, but overall you know that here what will be there? Medullary fluid or the interstitial fluid will be there. Now when you look at this thick descending limb, it is lined by simple. Whenever we use the word simple, means it is single layered. If there will be compound epithelium, that will be multi-layered. So lined by simple cuboidal epithelium, and this one, which is the thin descending limb. So this one is lined by flat cells, that is simple squamous epithelium. Okay, so this one upper part is cuboidal, lower part is what? This is the flat cells. This is, so here also the story is same. So this is thin part, this is thick part. So thick ascending limb, okay? Thick ascending limb. I told you about this or this. So same type of cells you will see here, okay? Same type of cells you are going to see here. Here also we'll say the simple cuboidal epithelium. And this is basically the simple squamous epithelium. That's it. And this loop of Henle, it's very important. It's along with vasa recta, your vas recta. It helps in concentrating the filtrate, or I would say now that filtrate will become, then after that filtrate will become urine. So beta, remember this. When you talk about the descending limb, I'm going here as per NCERT, right? Do remember it. In the this urine formation part, we'll remember everything. So descending limb is permeable to water. These are direct PYQs. Permeable to water, like water can move in and out, and impermeable to ions or electrolytes. And ascending limb here, permeable to ions but impermeable to water. So imagine your filtrate in PCT, it will be reabsorbed. So here if it is permeable to water, so actually the water comes from this part, especially from the thin segment here, especially from the thin segment, beta, right? The water will come out, and here in the thick part, this is the thin part of ascending limb. So here passively these electrolytes or ions, they come out, and here actively. But yes, so it will not allow the entry and exit of the water. So this is a very important point that you all need to remember, okay? Right, this is something very important that you need to remember. Please write down, quick. Notes are important here, okay? Okay? Sure? Okay, beta? Sure? So the next part is this DCT. So now this, yes, so next is the distal convoluted tubule. So you know that the ascending limb of loop of Henle merges in DCT, right? So this DCT, it is also present in cortex region. Then DCT is lined by just like that PCT, but here it is having simple cuboidal epithelium. Okay, next is here in this DCT, you will see DCT contains a specialized juxtaglomerular So it is special, it gets specialized from a specialized apparatus called juxtaglomerular apparatus. So we will discuss about it, that how with the help of this this arterioles it forms a juxtaglomerular apparatus, but this part we'll discuss later. But right now you just write the important points here, okay? Right, and here in DCT, conditional means when required. So conditional absorption or reabsorption. Conditional absorption of sodium and water occurs here. Okay, occurs here. So students, just remember this thing. This is your DCT, okay? This is your DCT. So it extends a bit. So this forms a kind of, not a kind of, actually a collecting tubule, but that tubule will further open into this part. That is your collecting duct. Okay, so many nephrons, right? They open together into the, right? Many nephrons, where do they open? They open in the collecting ducts, right? So many DCTs open in this collecting duct. So now when you talk about this collecting duct, right, what do you have to remember? See, this collecting duct, a part of it is in cortex and major part is in medulla. Okay, these collecting ducts, they pass to medulla by that medullary pyramids, right? By that medullary pyramids, and in renal papilla, many collecting ducts join and form duct of Bellini. So column of Bertini is different. This is the duct of Bellini. Please write down. Yes, please. Quick. Understood? So collecting duct is actually, a part of it is in cortex, rest is in medulla. So it is in medullary pyramids, okay? Right? So in the papilla region of medullary pyramid, they'll join, they'll form the duct of Bellini. Is that clear? Is that clear? Okay. So many ducts of Bellini opens in. So these, this duct of Bellini, it's a short, thick duct which is having all that collecting duct, है ना, or collecting duct. So this duct of Bellini open at tip of papilla and then open in, and then open in. Renal pelvis through calyces. So if you have attended the last class, then it will be very easy for you. So let me show you the diagram. See this. So this is the medullary pyramid or the renal pyramid. This part here, narrower part is the renal papilla. Okay, so actually this part is the renal papilla. So you will see that your collecting duct, right? A part of it is in cortex, rest it is in the medullary pyramid. So many collecting ducts, they'll join, they will form a short, thick duct of Bellini, which is open at this tip, and finally it will open in the renal pelvis because it is having the urine, and then obviously ureter will take that, right? So that is how it works. Is that clear? Is that clear? So the diagram part clear, everybody, right? Diagram part clear. So many nephrons together, they pass, they form this large duct of Bellini. Okay, so these duct of Bellini, they run through this medullary pyramid, open into calyces that will lead it to the pelvis. Okay, okay, sure. Any doubts? Any doubts? So diagram clear, topic clear. Which part is made up of which epithelium? Is that clear? So now when you talk about the collecting duct, just a minute. So see, individually I have explained you each and every part. Okay, so ye dekho PCT, then your loop of Henle, then your DCT, and then the collecting tubule and the collecting duct. Okay, so here we have mentioned that this duct of Bellini open at the tip of the papillae and then open in renal pelvis through calyces. Through calyces. So that's the route. So this collecting duct, yes, so collecting duct is lined by columnar and cuboidal epithelium in different regions. So basically region wise you will see this in different regions. Okay, done beta. And sometimes you will find that sometimes ciliated, it is ciliated also, this epithelium is ciliated also. So this is about the collecting duct, right? This is about the collecting duct. So if somebody will ask you that what do you find in the cortex region, so cortex region will have your woman's capsule, PCT, even DCT, part of, I'm not saying major, but yeah, part of collecting duct. What is there in medulla? Loop of Henle, obviously both the limbs of that loop of Henle, and major part of collecting duct. Is that clear? So rest you know that the glomerulus, efferent arterial, peritubular capillary network, and this is what you have to remember. So this is important, right? That is also a PYQ. They can ask you which region is, which part is present where. Understood? Understood? So before starting the physiology of excretion, we will talk about the juxtamedullary and the cortical nephrons and the blood flow. Yes, that is the main part. We'll also talk about the blood flow there. So this part is over. Yes, this part is clear. Please write down. Done, students? Okay. So write down blood flow in kidney. So obviously one thing that we know is that it is the heart which is the pumping organ. Yes or no? It is the heart which is the pumping organ, and in the right, it is the heart which is the pumping organ. So basically you will see that the aorta will, the heart will pump the blood. So aorta, it will carry the oxygenated blood. Right, it will carry what? It will carry the oxygenated blood. So aorta will have the oxygenated blood, and then obviously it is going to form what? Right, it is going to form what? It will form your renal artery, right? Because renal artery will carry that blood into the kidney. Then after that, afferent arterioles, because that renal artery will divide into arterioles so that each and every neuron can get this blood. Then there will be glomerulus, then efferent arterioles, and then we know that we have peritubular capillaries, and even vasa recta is also the capillary that is covering your loop of Henle. And then we know that what will happen? Venules will form, then they will open in the inferior vena cava, because from the lower part of the body, right, which vein is going to carry the deoxygenated blood from the lower part of the kidney? So we know that it is going to be inferior vena cava. But yeah, so there are venules, but obviously renal vein, then inferior vena cava, and then obviously heart again. So this side is having, like you'll see here, the deoxygenated blood, oxygenated blood. This is the blood flow. So here when you will see the diagram also, like let's say this is the vasa recta, it will have the oxygenated as well as the deoxygenated blood, then it will pass to the right. I'll show you the diagram, you'll get an idea there. So venules, then renal vein, then inferior vena cava. So from lower side of the body, it is the inferior vena cava which will bring back the deoxygenated blood from these organs to the heart, right? And upper part, superior vena cava. From the heart muscles, we know that we have the coronary sinus. Coronary veins are there, so we have the coronary sinus. So this part is clear. This part is clear. So this is basically the blood flow. Now we will talk about what? We will talk about the cortical and the juxtamedullary nephrons. Is that clear? Done? So diagram. So see this, firstly understand it here. Beta, you will see that two type of nephrons are there. See, first of all, they are showing the renal cortex here, renal medulla here, obviously the pyramid part. Now when you see this nephron, right, which is having the short loop of Henle, this is the nephron which is having a very long loop of Henle. So the one which is short, which is having short loop of Henle, this is the cortical nephron, and around 85% of nephrons are this. And this one having a very long loop of Henle, these are the juxtamedullary nephrons. So whenever there is the water scarcity in our body, so for the concentrating the urine, this is used. So in the juxtamedullary nephron, it will be covered with the vasa recta. Cortical nephron, here you will see the vasa recta either absent or highly reduced. Now look at the way they are placed. So if you will see, this is the fibrous capsule of that kidney. Okay, so you will see this part, PCT, DCT, woman's capsule. It is near to that surface part, near to that capsule part, and it is having very short, right, loop of Henle, which will even stay in, right? A little part of it, a very little part of it will come in the medullary region. But now when I look at the juxtamedullary nephron, so you can see at the junction of this medulla and the cortex, you will see woman's capsule, PCT, DCT, and a very long loop of Henle having well-developed vasa recta. And as I said, it is 85, so it will make the 15% of that. So these are the differences which we have to remember. So if somebody will ask you that which nephron will play a role in the concentrating the urine, then what will you say? Right? Right? Okay, sure, done. So this is about the juxtamedullary and the cortical nephron. So write down the differences here. So that's what you have. So juxtacortical nephron and the juxtamedullary nephron. Is that clear? Ready, beta? So very important topic it is. You can get question from this part as well. Right? So this one constitute around, right, this one constitute around 75 to 85% of total nephrons, and here you will see 15 to 25% of total nephrons. Next is that Malpighian. Now you know what is the Malpighian capsule. Near to kidney surface. And here Malpighian capsule, a Malpighian body is at the junction of cortex and medulla. Loop of Henle is short and mostly confined to cortical region or a part of it extend in medulla. Short part of it extend in medulla. Here loop of Henle is long and extended deeply into medulla. So people, are you making the notes? You should. Okay, you should write down. Done, beta? So next is peritubular capillary network is well developed. Not well developed. Here vasa recta is highly reduced or absent. And here vasa recta is well developed. So any doubt here in any word? Well developed. So this is about the two types of nephrons. Understood? This is about the two types of nephrons. Please make the notes here. Quick. Okay. So blood flow and all clear. So next topic will be your mechanism or the physiology, mechanism of urine formation or the physiology of the excretion. So I believe this is clear. So this is the diagram. You can see that. And now what are we going to do? We will discuss this part from the NCERT as well. Okay. We'll highlight the NCERT. Ready? We'll highlight the NCERT. So this column of Bertini and all clear. So let's read it quickly. So here you know that each kidney has nearly 1 million, right? So each kidney has nearly 1 million complex tubular structure called nephrons. They are the functional units, direct PYQ. So each nephron has two parts. What are that two parts? The glomerulus. So this is the tuft of capillaries that we have just discussed, and the renal tubule. So this is what you see in the case of the nephron. So generally in your paper they use the NCERT language to put the question. So you people can see this very clearly. So each nephron has two parts. One is glomerulus and another is the renal tubules. So glomerulus is the tuft of capillaries, right? Glomerulus is the tuft of capillaries formed by afferent arteriole. You know that afferent arteriole, it is wider, its diameter is more. So it is a branch of renal artery. Just now we have completed the blood flow. So I hope this part is clear. So blood from the glomerulus is carried away by the efferent arteriole. So we know that efferent arteriole is narrower. So there is a double-walled cup-like structure called the Bowman's capsule. So in nephron, two things, glomerulus and the renal tubule. So that renal tubule, how will it start? It will start with this cup-shaped structure which is double-walled. So it encloses the glomerulus. So glomerulus along with Bowman's capsule is called the Malpighian body or the renal capsule. Is that clear? So tubule continues further to form a highly coiled network, that is proximal convoluted tubule. Is that clear? That is proximal convoluted tubule. Is that clear? What is it? It is the proximal convoluted tubule. Understood? So then a hairpin shaped Henle's loop is the next part of the tubule, having descending and the ascending limb. So ascending limb will continue as another highly coiled tubular region called DCT. Any doubts? Any doubts? Quick. Done? So DCTs of many nephron open into a straight tube called collecting duct, right? Many of which converge and open into the renal pelvis. So here in the NCERT, no doubt they have mentioned that it will converge, but they didn't, and open into the renal pelvis. So pelvis word is used here, right? But through medullary pyramids in the calyces. So rest of the things are missing, but yes, you should know about this. So if this is the pyramid, you know that this part is known as the renal papillae, right? And here we have minor calyx, then major, and finally it open into the renal pelvis. Right, beta? So collecting ducts, they will join, they will form duct of Bellini, open into this minor calyx. So calyx is singular, calyces are plural, minor, major, and finally renal pelvis, and the urine will further pass to ureter and then to urinary bladder. So this part is very important. Afferent diagrams, you really need to observe them very carefully. So afferent arteriole, you can see this is wider, and this one is E for exit, it is narrow. So this difference here in the diameter is very helpful because it will maintain the pressure here. That will help in the filtration, and that is the topic now we have to discuss. So any doubt? Any doubt? Right? Any doubt? Beta, Malpighian tubules, you discuss in the case of insects, right? Tube-like structure present at the junction of midgut, hindgut, help in the excretion there. In the case of cockroach, we discuss Malpighian tubules. This is Malpighian body. If you will write it as Malpighian tubule, it will be incorrect. Understood? It will be incorrect then. Okay? Okay. So here you can see Malpighian capsule, PCT, DCT. Where are they present? They are present in the cortical region. I told you this is a PYQ. Loop of Henle dips into the medulla. So majority of nephrons, the loop of Henle is too short, extend only very little into the medulla. So they are talking about the cortical nephron, around 75 to 85%. So they are cortical nephron. So in some of the nephron, loop of Henle is very long, runs deep into medulla. This is juxtamedullary nephron. So they will help to concentrate the urine. Is that clear? Is that clear? So efferent arteriole emerge from the glomerulus. They form a capillary network, right? That is peritubular capillary. Then minute vessels will run parallel to the loop of Henle, form U-shaped vasa recta, which is absent or reduced in cortical nephron. So as per NCERT, we have covered everything. Any doubts? Any doubts? Right? So next is the urine formation. So you can give it the title, either you can call it as the physiology of the excretion or mechanism of urine formation, or simply the urine formation. So whatever you want to write, you can write it. Okay, you can write it. So let's discuss about the urine formation, having 3 steps. Glomerular filtration, also known as ultrafiltration. Second step is tubular reabsorption. And the third step is tubular secretion. That is it. These are the 3 steps that we have to discuss. And here again, we'll clarify NCERT, but in this part, you really need to understand extra point, extra pressures here. Understood? So you guys will have 5 minutes ka break only, right? Just 5 minutes break. That's it. And dinner break, you will get it around 9:30 or at 10. Right? We'll discuss this topic after 5 minutes. Quick. So let's start it, okay? So look at this diagram. Let's suppose if you are talking about this part, afferent arteriole, right? This is that complete glomerular capillaries. Okay, this one is the efferent arteriole. We know it very well. Efferent is wider, efferent is narrower. The glomerular capillaries. Okay, actually, this is the site. I'll explain it in detail, but firstly, listen to me. So, this is the place where the first step of urine formation occur. I already explained you this. Okay, now what's going to happen? See, here in the efferent arteriole, the blood will enter, of course. The blood will enter here. Efferent arteriole is narrow. So obviously there will be a pressure gradient here, but other than that, what you have to see, right? We'll write the points later, but firstly let's understand this particular topic. Just you know that so here we will talk about the filtration membrane, right? Filtration membrane through which this filtration will occur. You know that, one second guys. First step is ultrafiltration or glomerular filtration, or glomerular filtration. This is the first thing here. Now, where will it occur? The site is Malpighian body. Now you know what is the Malpighian body, your renal capsule. You know that what is it, right? Now here you will see the filtration occurs through, firstly filtration occurs through filtration membrane. Is that clear? Filtration occurs through. How will it occur? It will occur through the, yes please everybody, it will occur through the filtration membrane. That's what we have to discuss now. Is that clear? Right? Filtration membrane. Okay? Okay. So this is the filtration membrane, also known as glomerular capsular membrane. So this membrane contains what? Here, so filtration occurs through the filtration membrane. That includes, and if there is filtration or diffusion, you know that the membrane has to be very thin. So here you will see, the endothelial cells of glomerular capillary or glomerulus. Then you will see the epithelium of that, right? So endothelial cells of the glomerulus. Then you will talk about the epithelium of what? Epithelium of the inner layer of the woman's capsule, which is having the specialized cells. Is that clear? So first is endothelial cells of the glomerulus, right? Then obviously glomerulus, then obviously epithelial cells of inner layer of woman's capsule. Third is the basement membrane present in between this. Okay, so we already know that, let's suppose if I have to discuss it, we know that, so it is the endothelial cells, or you can simply call it as endothelium, right? The simple epithelial tissue of the capillaries. So here we know it already. This is the fenestrae, that is the pores here, and here we know that slit pores are arranged in an intricated manner. Slit pores are arranged here in an intricated manner, or podocytes are arranged here in an intricated manner, and we know that here also in this layer also we will see the slit pores. Yeah, for simple filtration pores also we can call them, right? You can call them as slit pores, or you can also call them as filtration pores. And now we know that there will be the basement membrane present in between. Okay, what do we have? What else do we have here? We know that the basement membrane is also present. So through this layer, the filtration will occur. Is that clear? Through this layer, the filtration will occur. As such, it is given in your book as well. So you guys can see this. We'll come to this part later. So the glomerular capillary blood pressure causes the filtration through 3 layers. Right now we are talking about these 3 layers. So endothelium of glomerular blood vessel, epithelium of woman's capsule, we know that particularly of inner layer of woman's capsule, and a basement membrane present between these, these 2 layers. Is that clear? A basement membrane present between these 2 layers. So epithelial cells of this, right, already beta, right, when we were talking about the structure, we have covered it there. So see, epithelial cells of woman capsule, they are called podocytes, arranged in an intricate manner so that they can create the minute spaces which we are calling as slit pores or filtration pores or the filtration slits. Is that clear? Is that clear? So the blood will be pumped here so finely, right, that you will see almost all the constituents of the plasma except the plasma proteins will reach there in the lumen of the woman's capsule. This is what the ultrafiltration is, right? Just say here, right? I'm not writing it because I have already explained this part previously. So here you will see if this is the arrangement, right? If this is the arrangement, we know that here we have these glomerulus and the basement membrane too. So this is the ultrafiltration. Here you will get the filtrate. You are not calling it as urine. It is still the filtrate. You are not calling it as urine. It is still the filtrate because still it has to undergo tubular reabsorption, tubular secretion, then it will become the urine. So here this filtrate, right, when you will look at this filtrate, so you will see it is having everything of that plasma except that plasma proteins. So this filtration here is basically not something selective, it is non-selective. Essential things are also present in this filtrate. But okay, fine, we got it, the membrane is there. So even in NCERT, they talked about the pressures, but they didn't explain it. So now we will talk about that pressures here. So let's understand this, right? Then we will write it. Let's understand this first. If there is any doubt, do let me know in the chat section. So we have afferent arterioles. We have efferent arterioles, right? You can see there is a difference in their diameter. Now blood will flow here. So because of this thing, the pressure will increase here. Okay, the pressure will increase here. So here we are going to talk about 3 pressures. One pressure, the blood pressure in the glomerular blood pressure we will discuss, which is quite high, which is going to favor the filtration. Then obviously here also you will see, ultimately students, what is this blood? Right, ultimately you know that what is there in the blood. We have the blood plasma and we have the formed elements. This is what we have in the blood, yes or no? We have the plasma, right, that pale yellow thing, and then we have the formed elements. So formed elements may we have your erythrocytes, your leukocytes and your thrombocytes, that is your platelets. And here in the plasma, we know that 90 to 92% is the water only, right? So other than that, we know that we have the plasma proteins. Here you will also find the waste. You will also see the ions here, the electrolytes here, right? The plasma proteins here. We know that albumin, right? Prothrombin, prothrombin, etc., they are present here. We know that. Right, these plasma proteins, when you talk about these plasma proteins, beta, they are obviously of the large size. So basically when this filtration is going on, because pores are present, right, we have just discussed it, pores are present. So when this filtration is going on here, right, so in this filtrate, you are going to see everything, right? So in this filtrate, except plasma protein, except the proteins, the plasma proteins, they cannot cross these pores. So except plasma proteins, everything present in plasma is here. Present in plasma is here. So obviously glucose, sodium ion, chloride ion, bicarbonate ion, potassium ions, etc., everything will be there, and particularly glucose. We cannot pass out that glucose in the urine, right? If glucose is present in the urine, that is an abnormality. It means our kidneys are not functioning well. That is glycosuria. That's a sign of diabetes mellitus. So obviously we have to reabsorb the things from this, right? The reabsorption is important because this filtration here, right? This filtration here is actually not selective, right? It is non-selective kind of filtration. It is just happening here, okay? Okay, due to the filtration pressure, it is just taking place here. Are you getting my point? So understand one thing before writing that pressures. So as I said, the diameter, right? There is a difference in the diameter, told you already, right? There is a difference in the diameter. So let's suppose if this is the case, there is a difference in the diameter, told you already. So there is the glomerular blood pressure, that is around 70 to 75 mmHg. So that will favor the filtration. That will favor the filtration. That is going to do what? That will favor the filtration. But other than that, blood is, as I said, having plasma and the formed elements. Definitely, if RBCs will start passing through the urine, so that's a sign our kidneys are not functioning well. So in normal condition, this is not going to happen. So ultimately, after this filtration, if here you are talking about the fluid will also start moving in the reverse direction. Why? Because here also you will talk about another pressure. So basically, one pressure that we discuss is blood, glomerular blood pressure, right? Blood colloidal osmotic pressure and capsular hydrostatic pressure. These are the three pressures which are present here, right? These are the three pressures which are present here. Is that clear? So it is the glomerular blood pressure, or you can also call it as hydrostatic pressure. So blood colloidal osmotic pressure and capsular hydrostatic pressure. Is that clear? Is that clear? These are the three pressure there. So glomerular blood pressure या glomerular capsular pressure, right beta? Blood colloidal osmotic pressure, we will write it first, understand it. Okay, so blood colloidal osmotic pressure, glomerular hydrostatic, not capsular, glomerular hydrostatic pressure, right? And then you have the capsular hydrostatic pressure. So what am I trying to tell you? I'm saying that when blood will flow here, due to this difference, here the pressure will be more. That is going to favor this filtration. That is going to favor this filtration. So I told you it is the glomerular hydrostatic pressure. It is the glomerular hydrostatic pressure. This is what we are talking. So around 75 mmHg, it can be in between 60 to 75 mmHg. In some books they directly mention it is the 75 mmHg. You know that it varies from body to body, but it will be in this particular range. Okay. Now, of course, it is getting filtered, right? As I said, everything except the plasma protein will pass out here, right? So that is why it is important for us to do the reabsorption. But before that, what is happening? Now the fluid is also present here. That will start moving in the reverse direction. Why? Because of this blood. Because of this what? Because of this blood colloidal osmotic pressure. We know that here we have the plasma protein. We understand the concept of osmosis. We know that in the osmosis, there is the movement of water. There is a movement of solvent from its higher concentration to its lower concentration. We know that basically this is what the osmosis is. So if there is a place where there is the high solute concentration, there will be less solvent. And if there is another place where there is high or I would say the cell which are separated by semi-permeable membrane, you'll see if there is less solute means more solvent. So water will move from more solvent thing to less solvent thing, means water will move from less solute thing to more solute thing. That's what I'm trying to tell you. We know it very well. So now what is happening here? The very simple thing, the fluid will start moving in the reverse direction. Why? Because we have this blood colloidal, simple you can say colloidal osmotic pressure. Okay, so it is around 30 to 32 mmHg. Okay, 30 to 32 mmHg. So here, because these proteins present here, they will also resist this filtration, right? They'll be like, nahi yaar, we also need the fluid. Why are you sending it there? It is like that. Are you getting my point? Then after that, we have capsular hydrostatic pressure. After that, what else do we have? We have capsular hydrostatic pressure. So when you talk about this capsular hydrostatic pressure, it is around 10 to 15 mm of Hg, right? It is basically due to the right? It is basically due to this filtrate, this fluid that is here in the lumen of the woman's capsule. So what we discussed about the three types of pressure. Firstly, I told you blood from the dorsal aorta, from the renal artery will come in the atrioles. Afferent and efferent atrioles, we can see that there is a diameter difference. The pressure will be created there. So that is the glomerular hydrostatic pressure. Glomerular blood pressure, it is whatever you want to say, but that will be more appropriate. Glomerular hydrostatic pressure, it is the one that is favoring the filtration, right? It is the one that is favoring the filtration, okay? It is the one that is doing what? That is favoring the filtration, okay? And these two, they are opposing it. So this is favoring, and these two, they are opposing the filtration. So now after this, we know that, right? Because here the filtrate, the fluid here, it will pass to the lumen. So it is having everything except the plasma protein. So the fluid present here, it will also start showing movement in the reverse direction. Why? Because of the osmotic pressure there, right? But still, it is not more than this glomerular hydrostatic pressure. So the filtration will occur for sure. And now the third pressure which is resisting this filtration is the pressure of this filtrate. So here in this filtrate, my dear students, here in this filtrate, right, this is in the lumen here. So it is the capsular hydrostatic pressure. So you people can see, right, it will also restrict the filtration. So now see, this is around, let's suppose if it is 30, if it is 40, 10. So total is 40, and if I consider 75, let's suppose, yeah, even if I consider 70, so you can see, right, you can still see that this glomerular hydrostatic pressure which is favoring the filtration is more than this, right? It is more than the pressure which is resisting the filtration. So for sure the filtration will occur. So that is how this ultrafiltration thing occurs here. Am I clear? That is how this ultrafiltration thing occurs here. Is that clear? Is that clear? Right? So this is what I have written here also. I have explained you also. So the effective, so now we will talk about the effective filtration pressure. So it is the effective filtration pressure that causes the ultrafiltration. So it is determined by three pressures. Number one, glomerular hydrostatic pressure, told you already, right? Blood colloidal osmotic pressure, told you already, right? It is due to, okay, then the capsular hydrostatic pressure. So here you can see glomerular hydrostatic pressure is the blood pressure, yes, in the glomerular capillaries due to the differences in the diameter of afferent and the efferent arteriole. Is that clear? Sure? Sure? So it is the main driving force that causes the filtration. It is 60 to 75 mmHg. Am I clear? Then comes the colloidal osmotic pressure. It is the osmotic pressure which is created in the blood of glomerular capillaries. It is due to the plasma proteins. So it resists the filtration of fluid from the capillaries. It resists the filtration of fluid from the capillaries. Is that clear? Then capsular hydrostatic pressure is there, which is caused by the fluid or filtrate that reaches into woman's capsule. It resists the filtration. It is around 10 to 15 or 18 mmHg. So now see the total net filtration pressure, right? So you can check the value. It will be somewhere around 10 to 25. You can just simply calculate it. Take it 75 minus 32 plus 18. Check it. What will be there? Yes, what will be there? It is around 25 mmHg. So that is what the net effective filtration pressure is. Is that clear? Is that clear? Am I audible? Am I audible? Done, beta? So these are the pressures. So they are not discussed in the NCERT, but you people should know this. Okay? You people should know this. Is that clear? So this is about the first step, right? This is about the first step. So next thing that we have to discuss is the reabsorption. Okay, so now this portion of NCERT, let's simplify that. So here in the NCERT beta, this paragraph is given. So when I'll start the regulation of kidney function, then I will talk about it. Before that, I will talk about it. This paragraph we will discuss at the end, then you will be able to remember that. Okay, done. So now what are they saying? Very important information it is. So it is the amount of the filtrate formed by the kidneys per minute, right? It is the glomerular filtration rate. Now what we have to discuss, we have to talk about the GFR. Especially in the old age, such kind of things happen, right? Just say, let's suppose if kidneys, like for kidneys, various tests are there, just when we check it, we go for the GFR also. If GFR is affected, beta, if this glomerular filtration rate is affected, if there is a problem in the glomerular filtration rate, it indicates whether our kidneys are working fine or not. Right, GFR, and it is associated with the blood pressure, blood plasma level, right, everything. So we will talk about it. So here you can understand one thing. What is that? That the amount of the filtrate, right, amount of the filtrate formed by the kidneys per minute. What is it? It is the amount of the filtrate which is formed by the kidneys per minute. In 1 minute, what is the amount of filtrate that is formed? That is beta GFR. That is what? That is GFR, glomerular filtration rate. Glomerular filtration rate. Matlab how much kidneys are making in 1 minute, the filtrate amount. So in a healthy individual, you are saying 125 ml per minute, that is 180 liters per day. But we are not excreting out 180 liters per day. The urine amount, though it depends on our water intake as well, so it is 1 to 1.5 liters per day, right? Though it depends on what? It depends on our water intake as well, right? But here in general, this is what we study. Okay, so this is the GFR. If there is any change in the normal value, right, it indicates there is an issue with the blood pressure, blood plasma level. Number one. Number two, from the NCERT, again I'm simplifying. So already told you what is ultrafiltration, definition. This part you have to follow, and how that filtration will occur, which pressures are going to favor it and which are going to oppose it, and what is the net effective filtration pressure. So here, what are they saying? That on an average, 1100 to 1200 ml of blood is filtered by kidneys per minute. So kidneys per minute, the filtrate that they are making is 125 ml per minute, that is GFR. And what is the amount of blood that they are filtering in 1 minute? That is 1100 to 1200, which is roughly 1/5 of the blood pumped out by each ventricle of heart in a minute. You know that on an average, our heartbeat is 72 times per minute. 72 times per minute. And we know that with each beat, 70 ml of the blood will be pumped. So if you multiply it approximately, I'm not saying total, approximately you'll get 5 liter. So 5 liter means 5000 ml. So you know that 1/5 of that, right? 1/5 of that will be roughly equals to this. So here you are talking about nothing but the cardiac output. Hey, they are saying, na, 1/5 of the blood pumped out by each ventricle of heart in a minute. So they are talking about nothing here. They are talking about the cardiac output here, and that is equals to heart rate, that is heartbeat per minute, multiply by stroke volume. So stroke volume, we know that it is the blood pumped by each ventricle in per beat. So that is 72 into 70, approximately 5 liter, 5000 ml, and that's how you're going to check it. So this value is justified. Are you clear with this part? Any doubts here? Yes. Done? Done, beta? Done? So this is the very first step of the Yes, please. This is the very first step here. So see, so comparison of volume of filtrate. I told you 180 liter per day filtrate will be formed. Urine releases around 1.5 liter, and I told you this depends on our diet, our water intake as well. So nearly 99% of the filtrate will be reabsorbed. Okay, and maximum reabsorption will be there from the PCT. So this is the next part that we have to cover, that we have to discuss. So any doubt so far? Yes, please. Any doubt so far? Quick. Yes, please. Done, beta. So next part, we know that what is there. This is the PCT, proximal convoluted tubule. What's that? It is PCT, that is proximal convoluted tubule. That is what PCT is. Are you getting my point? That is what PCT is. Okay, that is what PCT is. Is that clear? So now let's understand it. What exactly it is? It is the PCT, proximal convoluted tubule. We know that it is lined by, just now we discussed it. It is lined by simple cuboidal brush bordered epithelium. Simple cuboidal brush bordered epithelium. So if it is brush bordered, so it is having microvilli. It is having microvilli. So you know that this epithelium is in such a way that it helps in reabsorption. Plus here you will see that it is fully loaded with mitochondria. So this mitochondria, because it is the powerhouse of the cell, we know it very well. So they form the ATP, and that ATP supports what? That ATP also supports the active transport here, right? That ATP also supports the active transport. I hope you know what active transport is. So active transport means that by using ATP, the things will be absorbed against the concentration gradient. That is, the absorption will be there from their lower concentration to higher. Usually it is high to low, that is passive, right? That is passive transport. But when it is low to high, that is active. So ATP is used here, right? Against the concentration gradient it is moving. Am I clear? Am I clear? So mitochondria, what's that? That is the powerhouse of the cell. That is going to support, that produces the ATP that supports the active transport. So here you will see, right, the glucose, even the amino acids, even your sodium, even potassium, they show the active transport, right? Your chloride ions, water, urea, it shows passive transport. Am I clear? It shows what? It shows passive transport. So write down. Write down. Quick. Done, beta? Sure? Done. So this is what you see here. Okay, so this active transport of ions reduces the concentration of filtrate, of course we know it very well, है ना? And the equal amount of water is pumped into, because they are nearby. So that's why I told you the structure needs to be very clear. Is pumped into peritubular capillaries network by osmosis. Am I clear? By osmosis. So write down. Right? Write down. Quick. Guys, quick. Done, beta? So you can simply say that nearly all essential nutrients and 70 to 80% of electrolytes and water is reabsorbed here. Okay, beta, is reabsorbed here. Am I clear? So that is going to be reabsorbed here. Understood? Understood? Right? Thank you, Bhavani. Okay? Beta, glucose, amino acid, sodium, potassium, told you, right? Noted? Okay, so here it is not the complete urea, right? Some urea, because urea is our nitrogenous waste, right? Urea is the nitrogenous waste here. Okay, so if it is some urea, that will be reabsorbed, right? That will be reabsorbed passively. Rest will be there in the filtrate so that it can excrete out. Okay, so that's what you have to remember. Okay, so I hope this part is clear. So this is regarding your PCT part. Even in the NCERT, हाँ ये देखो, tubular epithelial cells, they perform the active or the passive reabsorption mechanism. जैसे glucose, amino acid, sodium, potassium, they are reabsorbed actively, means by active transport by using ATP against the concentration gradient, whereas the nitrogenous waste, they are absorbed by passive transport. Okay, so reabsorption of water also occur here in the initial segment. So this part is clear, because next we will talk about the loop of Henle. Okay, next we are going to talk about the loop of Henle, and about their secretion also we will discuss later. Okay, so when we were discussing the structure of the nephron initially, I told you about the loop of Henle. Yes or no? I told you about the loop of Henle. Yes or no? See, जैसे PCT में 70 to 80% of electrolyte and the water will be absorbed, then it will pass to this part. So you know that it is the descending limb, right? Now I'm not mentioning the details of, it is also not given in the NCERT. The thick part is absorbing this or thin this. Overall, you know that it is permeable to water. It is what? It is permeable to water, impermeable to electrolytes. I hope you know. This is important. So now you understand this part. When the filtrate will come out through this, filtrate is having solute, it is having solvent, and why this is happening, how will it occur, it will be discussed in detail in countercurrent mechanism, right? So have patience, listen to this. So you know that this part, right? We know that here in the surrounding we have the medullary fluid, and you know that this part is also covered with the vasa recta. Okay, this part is also covered with the vasa recta. Yes or no? Now this part is permeable to water, है ना? So water basically it is coming out. It is coming out. It is passing there in the medullary fluid. Then later it will pass in the surrounding vasa recta as well. That is a different thing. We will discuss it in countercurrent mechanism. Then you will understand it in a better way. So as of now, see this, right? This is the filtrate. Filtrate will pass through the descending limb. Obviously, if water is coming out, imagine a solution which is concentrated. Concentrated, like in the lab also, we talk about the diluted HCl or HNO3 or concentrated. So diluted, it is having more water, less solute. Concentrated, having more solute. So this filtrate, when it will pass through this, and you are just taking out the water, you're just taking out the water from it, so it is left with what? Solute, right? So obviously by the time it will reach here, you will see that the filtrate is going to be highly concentrated. The filtrate will be concentrated, like it will have very less water and it will have more solute, right? Now when that filtrate, when it will pass here, when it will go in the ascending limb, then what is happening? In this thin segment, you will see passively, right? Passively electrolytes are coming, your sodium, your chloride, they are coming. Here they will come out actively. By active transport, they will come out. है ना? So when this filtrate will pass through the ascending limb, right? Because electrolytes are coming, then again it will become diluted, right? So how? Here from that filtrate, you are taking out water. You are not decreasing or doing anything to the solute. You are just decreasing the water there. So water, if you are going to decrease, then also it is going to concentrate this. Now here, what are you doing? You're not adding the water here. You are just taking out the electrolyte. So this is what the reabsorption is, right? So by the time it will reach here, obviously its osmolarity, its solute concentration will again decrease. So in the DCT part, right? DCT part, there will be the conditional reabsorption. What will be there? There will be the conditional reabsorption. If required, water will be reabsorbed here, sodium will be reabsorbed here. If not required, it will not. Am I clear? It will not, right? So try to recall this concept, then we'll write it. Quick. Right? And beta, overall, if I have to tell you, right? Overall, if I have to tell you, just say you'll see that from the thin part majorly, you'll see that water will come out. It is said overall in NCERT, they didn't specify, they have simply mentioned that it is permeable to water, impermeable to electrolyte, but overall thick part, it doesn't even pass the urea or any other iron, yeah, any other, even not even the water. From the thin part, you'll see that absorption is there. Okay. Okay, so this part is clear. This part is clear. Quickly tell me, this part is clear. Done. So what exactly is happening? So in the loop of Henle, in descending limb, right? And if I have to be more specific, I would say thin segment of the descending limb. So in descending limb, what will happen in the descending limb? Descending limb is freely permeable to water. As I mentioned there, it is freely permeable to water. So imagine, right? It is your descending limb. As I said, how the water will come out and all that things, you will discuss that in countercurrent mechanism. I hope this is clear. So obviously it is the surrounding. It is in kidney, it is in medulla, surrounding medullary fluid. You can also call it as interstitial fluid. Done, beta? So what will happen? Here you will see more urea is there, right? What will you see here? You will see here the urea is more, right? Here in the interstitial fluid, beta, its solute concentration is more. Do you know that? Its solute concentration is more, right? And mainly, Solute concentration is more means osmolarity is more. So NaCl and urea, they maintain osmolarity. Actually, they maintain osmolarity. Osmolarity means solute concentration. So they are going to maintain the solute concentration here, right? They are going to maintain the solute concentration here. So this is something that you have to remember, right? That from the descending limb, we know that it is freely permeable to water, but why water will come out? Of course, again, the osmosis thing will be there. So water is coming out because the surrounding is more concentrated. It is having more NaCl and urea. So that is why water is coming out here. Got this? That is why water is coming out here. Is that clear? Is that clear? Right? So I am writing this key surrounding medullary fluid contain, please write down surrounding medullary fluid has high osmotic pressure. Due to high concentration of NaCl and urea. Am I clear? So due to high concentration of NaCl and urea. Is that clear? So water will be drawn out. Done, beta? Okay. Sure? Noted? So therefore, what is going to happen here? So therefore, water is drawn out. Therefore, water is drawn out and the filtrate become concentrated, or you can say hypertonic, when it reaches at the turn of loop of Henle. Noted? Please tell me, noted? Done, beta? Noted? Now we will talk about the ascending limb part. So firstly, write down quick. Done, beta? Okay, now next part is Now ascending limb is impermeable to water throughout its length, right? But is permeable to ions. Yes or no? Now what's happening? Now see, this is the ascending limb. Chalo, right? This is the ascending limb. You can see that. Guys, am I audible? So this is the ascending limb. So we know that it is impermeable. Already told you. This is impermeable to water, but permeable to ions. Now from this, your NaCl and all, they'll come out passively, right? They'll come out passively. But let me tell you here, in the ascending limb, please keep it in your mind, some amount of urea, just keep it in your mind, will enter also. Urea will enter also. And here I told you already that active transport will occur. So tell me any doubt. So the concentrated urine, when it will pass through it, when its electrolytes will be removed, ions will be removed. So don't you think filtrate will again become hypotonic? Yes or no? Yes or no? Yes or no? Yes, please. Okay. So here remember, here in this part, ions leave the filtrate, right? Even the potassium ion also. Ions leave the filtrate, but urea, small amount of urea, I would say, urea enters. So keep it in your mind. Please keep it in your mind. Okay. Just keep it in your mind that it's not like that if it is permeable or impermeable, then the things will only come out. Permeable means it is allowing them to cross. So it can enter also, it can leave also. Okay, it can enter also, it can leave also. Okay, okay. Done. So ascending limb is impermeable to water through its length, but is permeable to iron. So I told you that that narrow part, from here the ions will come out even, but the urea will enter. Is that clear? Is that clear? Sure? Sure, beta. So here you know that active transport, so here only they'll come out. Ions will come out here. Okay, so that is how it works. So you have to remember it. So ye dekho, so absorption, reabsorption is minimum in the ascending limb, right? So here you guys can see reabsorption is minimum in its ascending limb. So we will cover it, but after a short break. Okay, you can have your dinner break now. Okay, so then we will discuss it further. We will continue this part. Hi students, am I audible? If yes, then let's start. So yes, what were we talking? We were talking about the Henle's loop. Yes, so ascending limb part is done. So students, when it comes to the reabsorption, okay, so DCT, collecting duct or collecting tubule. So when plasma falls, when plasma level falls, then from posterior lobe of pituitary, right, then from posterior lobe of pituitary, that is your neurohypophysis, pars nervosa, ADH, that is anti-diuretic hormone, या vasopressin release, okay, and that makes DCT and collecting duct permeable and water absorption occur, okay, and water absorption occurs. Is that clear? So I'm repeating myself again. We know that filtrate will form, right? So in net effective filtration pressure is around 20 to 25 mm Hg. Filtration will occur. So 70 to 80% of the filtrate will be reabsorbed in the proximal convoluted tubule. Why? Because there you will see simple cuboidal. Simple means single layered, cube-like brush bordered epithelium. Brush bordered means having microvilli. That will increase the area for absorption. Then after that, it will further pass to the descending limb of the loop of Henle, which is permeable, especially the thin part, permeable to water, impermeable to electrolytes. So because in the surrounding, right, that's what I told you, because in the surrounding NaCl, urea is there, so the water will be taken out, and by the time the filtrate will reach here, it will be highly concentrated. Now from this side, that is the ascending limb, electrolytes will come out. So from this thin portion, sodium, chloride, even the potassium ion also come out. And here, remember one thing, the urea will enter. Urea will be reabsorbed here in the filtrate, and then from here also the ions will come out, the filtrate will be diluted. Now when it comes to the DCT, I told you already there in DCT, even in collecting duct, there is the conditional reabsorption, right? Conditional reabsorption of sodium and water. Conditional reabsorption of sodium and water. Is that clear? So why, when there is a requirement of their reabsorption, then only it will, like for an example, if in the plasma, if plasma fall down, right, the level of plasma fall down, then from posterior lobe of pituitary, this anti-diuretic hormone, which is ADH, which is vasopressin, it will release. Let me tell you this thing, this hormone is not synthesized by posterior lobe of pituitary, rather it is synthesized by hypothalamus, stored and released by this posterior lobe of pituitary. So it will make this collecting this DCT and the collecting duct water permeable, water will be reabsorbed, right? And from adrenal cortex, so it depends on the condition. So from adrenal cortex, aldosterone releases that help in sodium reabsorption when the solute concentration is less. So that is how it works. So it will reabsorb, right? So it is the conditional reabsorption that will occur from this part. So that's what you have to remember. So even in the NCERT, from the NCERT, we have revised it. So just have a look. Look at this Henle's loop, reabsorption is minimum. However, this region play a significant role in the maintenance of high osmolarity. So Henle's loop overall, minimum absorption, especially in the ascending limb, but it will maintain the osmolarity, that is solute concentration of the surrounding interstitial fluid, right? So descending limb, beta, permeable to water, but almost impermeable to electrolyte, almost impermeable, right? Pay attention there, right? Beta, in PCT, there is the maximum reabsorption overall, including water and the ions, right? So this concentrate the filtrate as it moves down. Ascending limb is impermeable to water, but allow the transport of electrolytes actively or passively both, right? So therefore the concentrated filtrate passes upward and it is diluted due to this electrolyte movement in the medullary fluid. So here conditional reabsorption under the hormones influence will occur. Okay beta, so DCT can also reabsorb bicarbonate. Do underline this part, do include it in your this part. Okay, so from DCT even the reabsorption of bicarbonate can also occur, right? And there is the selective secretion of hydrogen and potassium ion. So this reabsorption part is done. Now focus on the secretion, focus on the NCERT. So here dekho, this arrow is telling us that these things are getting added in the filtrate. So if I talk about the tubular secretion, so I will see that in the PCT, that's what you have to highlight also, H positive and ammonia, they are entered there in the filtrate. And here if you will see, this is potassium and H positive. So if you have very old NCERT, you'll find this data a little different. So this is what you have to focus. Am I clear? Am I clear? Please write down. So in the old NCERT, here also they used to mark the potassium, but that's what you have to emphasize. So look at the, see this. So H positive, K positive, ammonia into the filtrate. So text is telling us that there is H positive also, right? PCT mein H positive, K positive, ammonia. So tubular secretion is very important because it helps in maintenance of ionic and the acid balance. So let's suppose if we have more H positive in our body, so that will be passed out in the filtrate, that will be excreted out then. So it also maintains the pH. So as per the data, if you see, so H positive, K positive, ammonia, as per the theory I would say, and here what they have written, they have just written the H positive and K positive. Okay, okay. So, but in the diagram, they didn't mention the K positive here, though it is showing the reabsorption. So, both the things you have to keep in your mind, okay? Right? Understood? Right? So, now are you clear with this part? This is some amount of urea enters in the filtrate, but that urea is again reabsorbed by collecting duct. So, we will learn this thing. Right, we will learn this thing now. So this is all about the urine formation. Again here they have mentioned it that this collecting duct is a long duct which is extending from cortex of the kidney to the inner parts of the medulla. Large amount of water could be reabsorbed from this region to produce a concentrated urine. So this is very important for the concentration of the urine, right? So this segment will allow the passage of small amount of urea into the medullary fluid as well. So this also plays a very important role to maintain the pH and the ionic balance. Here also selective secretion of H positive and K positive is there. So as per the NCERT, I am specifying H positive, K positive, ammonia, H positive and K positive. As per NCERT, I am mentioning beta everything. NCERT theory part. Write down. Write down quickly. Clear? So next is the urine countercurrent mechanism. Okay, next is what? Next is the countercurrent mechanism. Okay, done. Done, bache? Okay. So now when you look at this diagram, so ultimately filtrate will pass here, right? Filtrate will pass here, filtrate will pass here, and when it will release from this collecting duct, then it is your final urine. Then it is the final urine. So remember this part, from the collecting duct also some amount of urea is diffusing in the medullary fluid, and from this thin part of this ascending limb, urea also enters in the filtrate. So it is the urea and NaCl. Right, now you will understand this thing. Urea and NaCl that maintain osmolarity of interstitial fluid. Understood? Sure? Okay, so next is the countercurrent mechanism. So students, I have seen that some students, they find it difficult, but trust me, it's very easy. So please pay attention, okay? So countercurrent mechanism. First of all, what is the meaning of countercurrent? Like flow is in opposite direction. This is the meaning here. Why is it used? It is used by higher vertebrates to concentrate the urine, right? To do what? To concentrate the urine and to conserve more and more water in the body. Okay, so what is the purpose of this countercurrent mechanism? To concentrate the urine so that water can be reabsorbed in the body, right? And obviously it is also the terrestrial adaptation, right? Now when you talk about the countercurrent mechanism, so it makes urine 4 times more concentrated than the filtrate. Okay, so if, right, if filtrate is, let's suppose 300 m osmol per liter, right, this is the unit of the osmolarity, then the urine will become, as I said, 4 times 12 m osmol per liter. That will be the osmolarity of the urine. So that is what you have to keep in your mind. Is that clear? That is what you have to keep in your mind. So now just look at this, right? Just look at this. Now I'm not making thick and thin part. You please pay attention, right? Here the juxtamedullary nephrons, they are going to help. So firstly, I'll explain it with the help of diagram, and then trust me, everything will be super duper clear from the NCERT only. So for this topic, you understand the concept the way I am telling you, and then just read it from the NCERT. So firstly, see this part. This is the descending limb of loop of Henle. This is the ascending limb. You know that here also the filtrate, the flow of filtrate is in the opposite direction. Means here if it is going downward, there it is going upward. It is like that. Yes, please. It is like that. So they both are in countercurrent. And now we know that, right? Now we know that U-shaped vasa recta is also covering it. U-shaped vasa recta is also covering it. Is that clear? U-shaped vasa recta is also covering it. Is that clear? Is that clear? Right, so this is the descending limb. This is the ascending limb. Right, and this one is also the descending limb. Right, vasa recta. So what's that? It is the vasa recta. So here you can see, here even in vasa recta is actually what? Vasa recta is actually the blood capillary, isn't it? It is actually what? It is actually the blood capillary. So if you see this thing, so in the vasa recta also, here you have the descending limb of loop of Henle. This one is the ascending limb of vasa recta. They are in the vicinity. So overall in vasa recta also, here the blood is moving upward, blood is going downward. But if you see, if you compare these two also, they are also in the countercurrent. So the flow is in the opposite direction. That's the first thing that you have to remember. Okay, the flow is in the opposite direction. That is the very first thing that we need to know. Okay, so if here it is descending limb and here it is the ascending limb, here it is the ascending limb, it is the descending limb. Now we know the conditions here. We know that this one is the impermeable. Yeah, I would say permeable to water, है ना? Impermeable to this line we are going to use multiple times. Impermeable to, I will write electrolytes here. And this one I know is permeable to, actively or passively, electrolytes. But impermeable to water. Now you forget about everything, okay? Let's not talk about the osmolarity. Let's not talk about any other thing. Just pay attention here. And here in the surrounding, because it is the juxtamedullary nephron that is helping here, right? That is going to help in concentrating the urine, right? It is the juxtamedullary nephron which is doing what? Which is basically, right? Which is basically doing what? Which is basically Concentrating the urine. Is that clear? Right? And obviously in this part, this complete surrounding, what exactly it is? It is the medullary fluid, na? It is the medullary fluid whose osmolarity is maintained by NaCl and urea. It is the medullary fluid whose osmolarity is maintained by NaCl and urea. Is that clear? I hope it is. I hope it is. So basically what is happening? From this side, water is coming out. They are not like we are permeable to this, we are permeable to that. They are okay with everything. Are you getting my point? They are okay with everything. This is what you have to remember. From this side, NaCl, from ascending limb, NaCl is coming out from the ascending limb of loop of Henle. It is entering in the, it is coming in the medullary fluid, then it is entering in the descending limb. And here, descending limb is releasing water in the surrounding. Right? That's what we have to remember. Please pay attention. That is what you have to remember. Understood? Understood? Now, here what will you see? Here what will you see? From descending limb, water is coming out in the medullary fluid and it will enter here in the ascending limb. And here, this ascending limb will throw out what? It will throw out the NaCl. That's what I'm trying to tell you. Right? That is what I am trying to tell you. Is that clear? Is that clear? Firstly, tell me, is that clear? How and why we'll discuss later. Firstly, descending limb, you know it, permeable to water. Water is coming out in the medullary fluid. That is entering in the surrounding blood vessel. That blood vessel is carrying it to the body. And blood vessel in return is leaving NaCl in the medullary fluid. Here, in the from the ascending limb, NaCl is coming in the medullary fluid. It is entering in the descending limb. And from here, water is coming out. Understood? And from here, water is coming out. Understood? Understood? So this is the complete thing here. So again, this one is throwing NaCl, this one is picking NaCl and releasing water. This one is throwing water in the medullary fluid, this one is picking that up, this one is giving the NaCl. That is what countercurrent mechanism is. Firstly, tell me this part is clear to you or not? Is it clear in your mind ki okay fine, theek hai ma'am, this is how the things are happening, yes or no? If it is clear, then do let me know. Ki yes ma'am, this part is over, we are able to understand that how does it work. Is that clear? Sure? Sure? Okay. So, see, actually what is happening? Why this juxtamedullary nephron is helping? What is the importance of this medullary fluid? Simple concept of osmolarity is going on. When your filtrate, when it enters in that PCT part, na, its osmolarity, you know that it is around 200 mmol. Slowly it will become 300. So by the time it will, this part will dip down here in the medulla. By the time this filtrate will move here, you know that what exactly is happening. The water is coming out. Obviously, the filtrate was becoming concentrated. In the last topic, we discussed that. And here, because electrolytes are coming out, the filtrate was again, you know, what was happening? The filtrate was again becoming the hypotonic. So this part you have to understand. Just imagine, this is the thing here. If from this side, NaCl is coming out, what is coming out? Let's suppose filtrate is here, NaCl, it is entering here in the medullary fluid. Right? Listen to me very carefully. NaCl from the ascending limb, it is coming out in the medullary fluid. Right? And this is what we already know. Now, if NaCl is getting entered here in the medullary fluid, overall, don't you think it will increase the osmolarity of that medullary fluid? If NaCl, that is if NaCl, if it is coming out from the ascending limb, it is coming out in the surrounding, that is in the medullary fluid, then definitely it is increasing the osmolarity here. So whenever the filtrate will pass through the descending limb, whenever the filtrate will pass through the descending limb, because in the surrounding osmolarity is more, it is having NaCl, it is even having urea, so that will definitely draw the water out of it. This is the concept of osmosis, right? Like let's suppose if there is one cell, this is another cell. If this cell is having more solute and less solvent, right, and this cell is having more solvent and less solute, then obviously we know that through a semi-permeable membrane, what will happen? Water. It is the water that will pass from this cell B to cell A. This happens in our body. So now, because NaCl is coming out from the ascending limb, either either by passively or actively, right, but it is increasing the osmolarity of the surrounding. So that is why water will withdraw from this filtrate. That is the reason water will come here. Now understand it in this way also. Now you must be thinking, ma'am, NaCl will also enter in the blood vessel. Yes, it will enter in the blood vessel, but it's not like the entire, everything from the, all the NaCl will enter here. Of course not. So, in the surrounding. Now, understand it in this way. In the surrounding, you have NaCl. That NaCl will be picked up by descending limb. Why? Because descending limb it has permeability for the both. So if here more NaCl, so then descending limb is like, okay, fine, osmolarity is increasing, chalo, I'll put some water there. To maintain that balance, I'll pick up the NaCl. Now when this will go here, here you know that water is coming in the surrounding. So that water will also picked up by the blood vessel here. So that water is basically entering, where is it entering? In the blood plasma. Where is it going? It is going in the blood plasma. So it will be carried away. And obviously because it is taking more water here, because here basically what is happening in the surrounding, if you will see, water is more. So just to balance that, NaCl will be released. So somewhere you can see that if NaCl is picked up by this blood capillary here, it is releasing it here only. It is not taking it anywhere. It is releasing it here only. Right? So it is picked up from this side, it is released from this side. When it is released, water is taken. When it is picked up, the water is released. So ultimately NaCl is not going anywhere. It is just roaming here and there. So this type of conditions, they are in our kidney, so that we can conserve more and more water. If we have to take out more and more water from this filtrate, obviously surrounding should have more and more NaCl, number one. Then rest of the things, so you know that in the DCT and in the collecting duct, as per the body demand, hormones, they'll do it. So this is what countercurrent mechanism is, right? This is what countercurrent mechanism is, and trust me, it's a very simple topic. So always remember, whenever you will talk about the descending limb of loop of Henle, so in its vicinity you will always find ascending limb of vasa recta and ascending limb here. So here you will always find descending limb of vasa recta. Is that clear? Is that clear? So when you look at this diagram of NCERT, majority of students, they just get confused, right? Majority of students, they genuinely get confused. They don't realize this part. See, it is, if it is like, you can just pick that up, you can just put it here like this, right? Just pick that up, put this here. So if it is descending limb, it is the ascending limb. Nearby to is ascending limb of vasa recta, and here to ascending limb, descending limb. So see this from this side, even in the NCERT it is given, water is coming out, NaCl is coming out. And from ascending limb side, NaCl is coming out and the water will be picked. And from this side, NaCl is entering, water is going out, the way I have explained. Right? Now look at the osmolarity also. So when this filtrate is here in this PCT part, 300 m osmol, the moment it is going down in the descending limb, because water is also coming out, 400 osmolarity, then 600, then 800, 1000, and by the time it reaches at this turn, it will be 1200 m osmol per liter, right? So this is the osmolarity of this inner medulla. So ultimately, like let's suppose if these are two cells, you know that if this is hypotonic, this is hypotonic, water will come here, right? And the conditions will be isotonic, right? This will happen, isotonic condition will be there, right? right? So iso means same tonicity, same solute concentration. So it doesn't mean that water movement will be restricted. If two cells, they have isotonic solutions, right? So the movement of water that is passing from this to this will be same, like this. Is that clear? Is that clear? Are you getting my point? Is that clear? So that's what you have to remember. So see, if here is 600 as water will come out by the time, water will come out at what level? Let's see if in the surrounding here, right? When you see deeply in the medulla, its osmolarity is 1200. So obviously the filtrate that is moving, it will give the water here. That water is immediately taken by this blood capillary. It is also releasing the NaCl here to maintain this osmolarity of the medullary fluid. So basically what is happening? So water will come till this filtrate and the surrounding osmolarity, they reaches at the same level. Now again here we are moving, we can see this. So basically even due to this osmolarity in the surrounding, water is also reabsorbed from the collecting duct. So collecting duct, from collecting duct, some amount of urea is also reabsorbed, and we know that some amount of urea is also secreted here in the filtrate by this thin part. That is what we have discussed. Is that clear? Is that clear? Quickly tell me, is that clear? So that is what countercurrent mechanism is, and the excess of reabsorption from DCT and collecting duct, it depends on the body needs. That we will understand in the next topic, that is regulation of kidney function. Am I clear? Am I clear? Okay, so see this image again. Water is coming out, है ना? Water is coming out. NaCl is coming out, and here water is coming out from this blood vessel, and NaCl is entering. NaCl is leaving, water is entering. So that water, when it will enter in this, right, ascending limb, it will be taken back into the body. That water is basically reabsorbed. Okay, so that is how it works, beta. There is one more diagram, if I'll get it. हाँ, ये देखो. See this, please. This contain everything that I have explained. Please read this part. Quick. Quick. Guys, quick. Noted? Okay, so now let's read it from the NCERT. So mammals, they have the ability to produce a concentrated urine, right? So Henle's loop and vasa recta, and we know that it is well developed in the case of juxtamedullary nephron, so they will help there. So Henle's loop and vasa recta, they play a significant role in this. So flow of filtrate in these two limbs also, we know that they are in the opposite direction. Even the flow of blood in the vasa recta is also in the opposite direction. Done. Now the proximity means closeness between the Henle's loop and vasa recta, as well as countercurrent in them, helping in maintaining and increasing the osmolarity towards the intermedullary interstitium. So I told you inner medulla is having the osmolarity of 1200 mmosmol per liter. So from the starting, right, from the cortex region, you will see it is 300 mmosmol. In the starting itself, that's why I told you filtrate is this, urine will become this, right? So it is 4 times. This is the 4 times of the filtrate. Is that clear? So gradient, how this osmolarity gradient is maintained in the medulla, this is caused by NaCl and urea. Again a PYQ multiple times, even I have repeated it here. Am I clear? Am I clear? So next part is कि NaCl is transported by the ascending limb of Henle's loop. NaCl is transported by the ascending limb of Henle's loop, that will be exchanged with the descending limb of vasa recta. So if this fact is clear in your mind, ascending limb of your loop of Henle is having descending limb of vasa recta in its vicinity. So you know that one will put NaCl in interstitium, another one will pick that up. So this NaCl, we know that it is returned to the interstitium by the ascending portion of vasa recta. Am I clear? By the ascending portion of vasa recta. Is that clear? Is that clear? So similarly, small amount of urea enter the thin segment of ascending limb of Henle's loop, that will be transported back to the interstitium by collecting tubule. Is that clear? So the above described transport facilitated by special arrangement of Henle's loop and vasa recta. So this is beta countercurrent mechanism. So this mechanism help to maintain a concentration gradient, right? And this diagram, just make it, it will explain everything. You will never ever get confused even in the future. Okay, so in the medullary interstitium, so presence of such interstitial gradient helps in an easy passage of water from the collecting tubule, thereby concentrating the filtrate. Right, so this is it. This is about this topic. So the next topic that we have to discuss is regulation of the kidney function. Am I clear? Next topic that we have to discuss is the regulation of the kidney function. Done, beta? Done? Done, beta? So this is about the countercurrent mechanism. I hope you are done with it. Okay, just revise it. Take 2 minutes and revise it. Okay, so next is the regulation of the kidney function, in which very first thing that we will discuss is, before that we'll talk about the juxtaglomerular apparatus. Okay, done? Any doubts? Okay, चलिए. Next is the regulation of kidney function, but before that, what are we going to talk about? We are going to talk about the juxtaglomerular apparatus. You remember in the initially I told you that forget about this paragraph, we'll discuss it later, है ना? So that is what we are going to talk about, juxtaglomerular apparatus. Very important, that will help in maintaining the glomerular filtration rate. Is that clear? Right? Is that clear? So it is the JGA apparatus. Right, it is the JGA apparatus, that is juxtaglomerular apparatus. Right, juxtaglomerular apparatus. Is that clear? So here it will include JGA apparatus. It will include what? It will include your juxtaglomerular cells, and they are also known as your granular cells plus macula densa, and not given in NCERT, but mesangial cells also. So they make your juxtaglomerular apparatus, right? They make your juxtaglomerular apparatus. Am I clear? What do? They make the juxtaglomerular apparatus. So that's what you have to remember. Now, what is this apparatus? What is its role? Let's discuss it with the help of this particular diagram. You remember this? So now here, if you will see, can you see that? See, this is like this diagram. This is the DCT, distal convoluted tubule. In the starting of the lecture also I told you about it. This is the DCT. So this DCT, it is in association with these arterioles here. Can you see that? They are in association with this arteriole here. Can you see that? Can you see that? It is in association with this arteriole here. So here, the part of DCT, can you see that this is distal convoluted tubule? The part of DCT which is coming in contact with these arterioles, you will see, right, with these arterioles, particularly afferent arteriole, you'll see the cells of this DCT, they are also getting modified. That modified cells of DCT are macula densa. Understand it from the image first. Now when you look at the afferent arteriole here in this wall, can you see some cells which are granular cells, which are also known as JG cells? And other than that, these cells are also present. These are mesangian या extraglomerular cell. So this is the complete juxtaglomerular apparatus, right? I will show you another diagram also, right? Firstly, just get this clear in your mind. Distal convoluted tubule. If you think, ma'am, how the DCT, you know that in one kidney you have around 1 million of neurons. It's not billion, it's million. So something like that. That is the sort of arrangement. And here we already know this, right, atriole. So DCT may the cells will get modified, you will call them as macula densa. That's what I'm trying to tell you, right? And here in this walls, you will see that you will have what? You will have the, see this. This is macula densa, right? And here in the walls, you can see that at the point where it is coming in the contact with these afferent arteriole, there you can see that the cells are modified. So now these juxtaglomerular cells, we'll write about them, right? They will form renin. We'll talk about it. Firstly, you write the points here. Okay. Okay. So juxtaglomerular apparatus is basically help in maintaining glomerular filtration rate, number one. So it's a special sensitive region formed at at the contact of DCT and afferent atriole. Understood? And afferent atriole. Is that clear? Right? Formed at the region where DCT and afferent atriole they contact. Is that clear? Is that clear? So the walls of arteriole, where they come in contact with right, when they come in contact with DCT, get swollen and contain cells having granular cells and form granular cells also called juxtaglomerular cells, juxtaglomerular cells. Am I clear? So these cells contain renin. So beta, it is single N wala renin. It is RENIN. What's that? It is RENIN. This is that renin. See, in the stomach, right? In case of infants, we see there is a formation of prorenin. That is RENIN. This is an inactive enzyme. This is a zymogen. Okay. But in the kidneys, what do you see? It is renin. So this is the enzyme here. Right. This is a hormone, protein, RENIN. It is RENIN. It is the single N. Okay. It is what? It is the single N. Am I clear? What's that? It is the single N. Is that clear? Is that clear? Quick. Right? Sure. Chaliye. So this is the juxtaglomerular cell. So got it? So it is RENIN, right? If you will write it RENNIN, then it will be incorrect. Okay? Okay. So smooth muscle cells of, right? Okay. Oh, I missed that part. Smooth muscle cells of, right, walls of arteriole. So smooth muscle cells of walls of both arteriole where it comes in contact with DCT, right, get swollen and they form these granular cells. So these are the juxtaglomerular cells. So these cells contain inactive renin. These cells contain inactive renin. Is that clear? So next is the macula densa that you have to write. So macula densa में cells of DCT epithelium comes in contact with arteriolar wall, right? Right, and they will get modified and they'll form the macula densa. So cells of DCT that comes in contact with arteriolar wall, get modified and then form macula densa. Matlab that cells are basically known as macula densa. So write down quick. Done, beta? Clear? Chalo, write down quick. So this is the juxtaglomerular apparatus. So let's mark it from the NCERT also, and then we will start the regulation of the kidney function. So here you can see the kidneys are having the built-in mechanism for the regulation of glomerular filtration rate, right? So it is carried out by JGA, which is a special sensitive region which is formed by cellular modification and DCT. So DCT mein that modification will form macula densa. Afferent arteriole mein, right, there are the modifications particularly in the smooth muscle cells of that arterioles. There you know that will be the juxtaglomerular cell. So when there is a fall in the GFR, it will activate this, right? So here they have mentioned JGA, but that regulation of kidney function, this is what they have mentioned here, okay, this part. So we will make a flow chart for it, okay? We will make what? We will make a flow chart for it, right? Then you will be able to understand it in a better way. So basically when there is a fall in the blood pressure, that will also, even if blood plasma is less, right, even if plasma level falls, if there is blood pressure is down, ultimately all that things, they affect the GFR, right? Ultimately all that things, what they do, they affect the GFR. So when the glomerular filtration rate is less, so this is going to activate this juxtaglomerular apparatus, and due to which what will happen, beta, the JG cells, they will release renin. What will they do? They will release renin. Now in the blood, right, just say liver, you know that liver, it forms, right, I'll make it properly, but right now you note down, it forms a plasma protein called angiotensinogen. Obviously it is inactive. Angiotensinogen, it is inactive, right? It is inactive. So now when the GFR is less, so this renin, it will act on this angiotensinogen. This is inactive plasma protein synthesized by what? Synthesized by the liver. So it will act on it, and what will you see? You will see that angiotensin 1 will form. Can you see that? Right? This is the active form here. Okay? Okay? So when blood pressure is down, blood volume is down, right, even your GFR is down, because overall, why is it happening? Because there is the less concentration. You can even write the reasons here. There is a less sodium concentration in, right, in basically what? In distal convoluted tubule. So all that factors are going to stimulate that. Is that clear? Yes, please. Done, beta? Done? So now this is also an inactive hormone. This is also not that active. So this angiotensin 1 will be converted to angiotensin 2. So actually this angiotensin 2 is the active one. Wait. So this renin, and so here I am writing. So angiotensinogen which is inactive plasma protein synthesized by liver. So what will happen here? So renin will act on it, and this is going to form what? Angiotensin. So this angiotensin 1 is also what? It is also inactive, right? It is not even, though it is angiotensinogen will get converted into the angiotensin 1, right? So this is inactive, this is active, but still its more active form is your angiotensin 2. So how will it form? So there is an enzyme here, that is ACE in lungs. So this in lungs epithelium is going to form it. So what is this ACE? It is angiotensin converting enzyme, right? It is angiotensin converting enzyme. So this angiotensin beta, this angiotensin 2 is also, right? It is also a powerful vasoconstrictor, right? Remember this, it is also a powerful vasoconstrictor. It also act on the PCT. It will constrict that, increase the blood pressure. I'll write that part later, but the main function is this angiotensin 2 will act on adrenal cortex. You know that when you talk about the kidneys, right? In the case of kidneys, we know that the cap of the kidney, what do you call it? Adrenal gland. Which gland it is? It is the adrenal gland. Yes, please. Which gland it is? It is the adrenal gland. So adrenal gland is having adrenal medulla and adrenal cortex. So when you people talk about the cortex part, so cortex is having 3 layers, right? For that layer, the trick is GFR, right? Right, Gaffer, GFR is the trick for that. That is outer one is zona glomerulosa, zona fasciculata, zona reticularis. This is the name of the layers of the adrenal cortex. Adrenal cortex, different layers, they'll release different hormones. Are you getting my point? Different layers, they release different hormones, students, right? Different layers, they will release different hormones. So here in this particular topic, which hormone do we need to discuss? We have to discuss the hormone, right? We have to discuss the hormone aldosterone. Are you getting my point? Which hormone you need to discuss here? You need to discuss the hormone aldosterone here. Is that clear? Sure. So this aldosterone is released by this zona glomerulosa, right? It is going to secrete this aldosterone. Understood? It is going to release this aldosterone, and I already told you that aldosterone will show the conditional reabsorption. So here you will see it will act on the adrenal cortex. So if at the place of cortex medulla is written, that is also incorrect. So in response to that, your aldosterone will be released. So it will act on distal part and DCT. It will act on the collecting tubule. It will help in the sodium reabsorption. Sodium will be reabsorbed. Water again will be reabsorbed. Is that clear? So this is basically the RAAS mechanism, that is renin, angiotensin, aldosterone system. So it is basically done to increase the blood pressure, right? It is basically done to do what? To increase the blood pressure. Firstly, write down that. Okay? Done? Done, beta? So as I said, this angiotensin 2, it's a powerful vasoconstrictor. That is, it constricts the blood vessels, right? So it acts on proximal part, right? It acts on proximal part of nephron. Are you getting my point? So when it is going to constrict the blood vessel, so obviously it will increase the blood pressure. It will indeed increase the blood pressure. Yes or no? Yes or no? Yes or no, beta? Done, beta? So this aldosterone, it will act on distal part, okay, due to which what will happen? Sodium reabsorption. So sodium, if sodium is reabsorbed more, then in response to that, because osmolarity will increase, water reabsorption will also occur, है ना? Blood volume and hence the blood pressure increases. But if there is sodium reabsorption, the potassium excretion will increase. Sodium reabsorption will be there and potassium excretion will increase, right? So obviously due to which there will be increase in blood pressure, increase in blood volume, and overall we know that there will be increase in GFR. This is what the RAAS mechanism is, right? This is what the RAAS mechanism is. Okay? Okay? We'll talk about the ADH also, but firstly tell me, this part is clear? Sure? Beta, sure? Quick. Quick. So firstly, let's see these images and then we'll read it from the NCERT. Dekho, drop in blood pressure, drop in fluid volume, that is going to send a signal. Your juxtaglomerular cells, they will release renin, right? They will release renin. That will act on angiotensin. So it is the inactive plasma protein made by liver, right? It is the angiotensinogen is written. Obviously, it is inactive plasma protein, right? Who synthesized it? Liver. Then you will have angiotensin 1, right? Right, then it will form angiotensin 2. Who will form? ACE. So it is angiotensin converting enzyme. It is released from the lungs. So ACE act on angiotensin 1. It will form angiotensin 2. That will act directly on the blood vessel, on the peripheral part, even on the, because vasoconstrictor act on blood pressure, stimulate the vasoconstriction. Right, beta? And this angiotensin 2 will also act on adrenal cortex, zona glomerulosa, which will release what? Aldosterone. That will act on the kidney, reabsorption of salt and water, and excretion of potassium will be there. One more elaborated part here you guys can see, right? So angiotensin 2, keep it in your mind, increases the sympathetic activity. So it increases the tubular sodium chloride reabsorption, definitely from the proximal part. So if sodium is reabsorbed, your potassium excretion will be there. So more sodium reabsorbed, water retention occur. Water retention. So that is why, just some people say na, ki we have the water retention. So that is why we immediately gain weight or we lose weight even if we start exercise. So in that case, doctors advise to take less sodium chloride, the less salt, because salt is ultimately having what? Salt is ultimately having the sodium chloride, right, beta? So here you will see, right, this angiotensin 2 will act on adrenal cortex, aldosterone secretion will also do the same, plus angiotensin 2 is vasoconstrictor, increase the blood pressure. Even this angiotensin 2, right, see how everything is related. So this plus is telling me that it is giving a like positive feedback that pituitary gland, please release the ADH. So angiotensin 2, this is the perfect flowchart for you. It also, right, this angiotensin 2, it will give the stimulatory signal for what? Right, for the secretion of the ADH. So ADH will give that signal to collecting duct so that water can reabsorb. So ADH is anti-diuretic hormone. What is this ADH that we are talking again? It prevent, anti-diuretic means it prevent diuresis. What is diuresis? What is diuresis? right? It prevent the diuresis. What is diuresis, beta? What is diuresis? Prevent diuresis, excessive diuresis is excessive urination. So it will prevent that. Okay, it will prevent that. Done. So that is how it works. So sodium, when it is reabsorbed, potassium excretion will occur. Okay, potassium excretion will occur. Done. Sure. So look at this part, right? So osmoreceptors, where are they present? In the body, in the hypothalamus, osmoreceptors are there. They can detect the osmotic balance, beta. So, right, they are activated when there is a change in the blood volume, in body fluid volume, and even in the ionic concentration. So when there is the excess of loss, right, excess of loss of fluid from the body, it can activate these receptors, and once they are activated, they'll stimulate the hypothalamus, because it is the hypothalamus, beta, that has formed the antidiuretic hormone, or vasopressin. Vasopressin means it will press, it will constrict blood vessel. That's the meaning here. It will constrict blood vessel. Am I clear? That it will do what? It will constrict the blood vessel, right? So we know that hypothalamus is going to make it. It will be stored in the neurohypophysis. So when there is a change in the body fluid volume, the osmoreceptor will tell hypothalamus that release this AD. Hypothalamus will tell posterior lobe of pituitary to release this ADH. And ADH means antidiuretic hormone. It facilitates the water reabsorption from the distal convoluted tubule, from the collecting duct part, and the diuresis will be prevented. That is, excess of urination will be prevented. Am I clear? So an increase in body fluid volume can switch off these osmoreceptor, and obviously the ADH release will also be decreased. And even I told you, when this angiotensin 2, it will also send a signal for the release of this ADH. Am I clear? Am I clear? So that is how collectively they maintain the GFR. So that is how the blood pressure is increased. But there is one more thing that we all need to understand, and we all need to take care. Done? What is that? That if there is an increase in blood pressure, we have to maintain it as well. There should be a check on that. So who's going to keep a check? An ANF, that is atrial natriuretic factor. Right? This is very important. It acts as a check on RAAS mechanism. It acts as a check on RAAS mechanism. Is that clear? It acts as a check on what? Check on RAAS mechanism. So ANF can cause vasodilation, that is dilation of the blood vessel, thereby decrease the blood pressure. Is that clear? Is that clear? This is something very important. Direct PYQ it is. So do remember it. So the next topic that we have to discuss is micturition. Then we will talk about the disorder. So I'll take hardly your 30 minutes and the chapter will be over. Understood? Chapter will be over then. But before that, just say, let's see. If you take too much of tea, coffee, right? We feel like urinating, After some time, we feel like urinating, isn't it? Isn't it? Yes, please. Done? Sure? Done, beta? So when we take tea and coffee, like we have that urge to urinate later. Why is it so? Because when you take these beverages, right? If beverages are there in our body, matlab body fluid volume is also maintained. Na? So basically when we take such beverages, they basically tell the hypothalamus that ADH secretion is not required much, it will be less. Just those who are alcoholic, right? If somebody take beer, whiskey, like that, they go to the they urinate a lot. Why is it so? Because that type of things, they signal the brain that less ADH need to be released, and already body fluid volume is more. So this coffee, tea, even the alcohol, all of that, what they do, they cause the less release of ADH. So less release of ADH matlab, if there is less ADH, it means more urination, right? It means more urination. And if there is more ADH beta, it means less urination. Am I clear? It means less urination. So that's what you have to remember, okay? Right? That is what you have to remember, okay? Right? Yeah, exactly. Because that is why these type of things, excellent beta, Umarumi. So these are the diuretics, right? Because they will cause that urination thing. Am I clear? Right? Sure. So one more thing, just see, if there is the hyposecretion of ADH, it can result in diabetes insipidus. Do you know that? It will result in diabetes insipidus, where that person will have excessive urination, person will have polydipsia, excessive thirst will be there, right? And obviously, because if there is more urination, the all that essential ions, they will also release, है ना, excessively. Done? Done, beta? So polydipsia can also occur. Right, beta? Okay. So excessive thirst, that will result in what? That will result in polydipsia, right? So polydipsia means that person will feel dehydrated, right? That person will sense the dehydration because there are high blood solute level, right? It can, like, both the things can initiate that. If there is excessive dehydration or high blood solute level, that person will feel thirsty, that person will demand water. Is that clear? Right? So hypersecretion of ADH, hyposecretion of hyposecretion of ADH will cause diabetes insipidus. And when you talk about the diabetes mellitus, right, mellitus, so here you will see there are two things here that we talk about a lot: glycosuria, even ketonuria is also because of that, right? So here what is happening, beta? When there is glycosuria means glucose in urine, so your kidneys are not functioning well. It is associated with the ADH secretion. Here your kidneys are not functioning well, so water soluble ketone bodies are found in urine, so they indicate diabetes mellitus, and the hyposecretion of ADH indicates diabetes insipidus. Am I clear? Sure. Sure. Chaliye, now let's complete the last part here. So let's do it. Okay? So we are about to finish it. So done. Chaliye, let's have 10 minutes ka break. Let's meet at 11:10. Okay, let's have 10 minutes ka break. Hi everyone, so I hope now I'm audible. Okay, so next is micturition. So here you people can see the urination is basically the micturition. So we know that this urine will be carried to the urinary bladder, right? It will be stored there unless the voluntary signal is given to the CNS. So already in the last part we talked about it. We know that the outer sphincter, which is at the neck part of the bladder, that is having the that is basically having the striated muscles, right, which are basically voluntary. Inner sphincter is having the smooth muscles, which is involuntary. So firstly, the voluntary signal will be passed to what? It will be given by the that signal, voluntary signal is given by the CNS. So this signal will initiate what? In the urinary bladder, there are the stretch receptors as well, right? You can see that. So when this urinary bladder is filled, it is going to, right, do what? It will send a signal to that stretch receptors, and in response to that, they will send signal to the CNS. Okay? They will send signals to the CNS. Is that clear? Is that clear? So CNS will passes on motor message, beta, right? CNS will passes on the motor message, so that what can happen? So that contraction of smooth muscle of bladder can occur, and this contraction of muscles will relax that sphincters. So I told you these sphincters, they remain contracted, right? So the urinary bladder is closed. When the sphincter, so this thing can confuse you in exam, so please keep it in your mind. So when the sphincter, yes, please. So when the sphincter muscles, when they are relaxed, right, then what will happen, beta? So sphincter muscles, they will relax, the urinary bladder will open, the urine will be released. So this is the micturition or the urination. And remember this thing clearly. Please remember this thing clearly, that it is the neural mechanism, okay? Okay, so it is a micturition reflex. And other than that, right, on an average, a healthy individual can excrete 1 to 1.5 liters of urine per day, right? It is light yellow colored, watery fluid, slightly acidic. But these things can vary. It depends on our diet, our water intake as well. And it is having a characteristic order. So 25 to 30 gram of urea is excreted out per day, my dear students. And various conditions can affect this thing. And when we analyze the urine, we can also test the malfunctioning of our kidney. Understood? So you know that if there is glycosuria and ketonuria, right, that is the indicator of diabetes mellitus. Multiple times they have repeated this question in the NEET examination. Is that clear? Is that clear? Sure? Done, beta. So now here they have mentioned now that our urine is having a characteristic odor. So why is it so? Why that odor is there? The reason is, you know that our excretory product is this urea. So when the urine is passed, so this urea will be converted to ammonia. So that ammonia is having the strong smell, right? That's why the urine, which is, you know, even very old, that is having more of the smell, because this ammonia itself is having that strong smell. Is that clear? Okay. Done, students. So next is the role of other organs in the excretion. So it is very simple. It is very, very simple. Our lungs, they remove the large amount of CO2. So this value, it should be clear in your mind, right? It should be clear in your mind, approximately 200 ml per minute. Okay. So liver, the largest gland in our body. So it is the largest gland. But if somebody will ask you the largest endocrine gland, then it is the thyroid. So it will secrete bile containing substances that are having, you know, these pigments, bilirubin, biliverdin, cholesterol, degraded steroid hormones, vitamin and drugs. And that is why even our excreta is having that characteristic color due to these pigments, right? So other than that, you can see the sweat and the sebaceous gland in the skin, they also eliminate, right, student? They also eliminate the waste. So sweat, right, produced by the sweat gland. It is the watery fluid which is having NaCl. Even it also contains small amount of urea, lactic acid as well. Understood? So sweating, it helps, it basically cool down the body surface, right? We have studied that in class 9th also. So sebaceous gland, that is oil glands, they eliminate the substances like sterols, hydrocarbon, waxes in the form of the sebum. Okay, and this sebum will provide a protective covering to our skin as well. So that's what you have to remember. Okay, okay, done. Yes, please. Beta, we also start statement based questions. We also start diagram based question. I don't think that Mr. IM 14 something something, you don't follow our channel sincerely. Huh? Chaliye. So next is disorders. So uremia means if there is the accumulation. So here I'll stick to the NCERT part. So here this is the accumulation of urea in blood, that is uremia. So if there is excess of, right, if there is excess of urea in the blood, that can result in the kidney failure. Okay, so if kidneys, they are not working, right, if kidneys, they are not working, then what is there be done? Hemodialysis will be done. Okay, hemodialysis will be done. So here what will happen? Let's suppose if this process is a blessing for the kidney patient. Now let's suppose, beta, what is the thing? That it is also known as artificial kidney, right, or hemodialysis. No doubt it is draining process as well, right, financially and financially also, right, it is a draining process as well, but still it is a blessing for the kidney patients, right, those who's the patient in their case, basically this technique, it's helped them for their survival. Are you getting my point? Yes, are you getting my point? Yes, beta. So artificial kidney, on what principle it is based? So basically it is the hemodialysis. Okay, so here you will see the main part will be, beta, dialyzer, right? You write down, I'll discuss the principle and everything will be with you will see the dialyzing fluid is also there, and you will also see what else do you need? You need a dialyzer, you need a dialyzing fluid, and you need a blood pump. Is that clear? Is that clear? Yes, please. You need a blood pump. These are the three main things that we need here. Actually, what is it? Right? What is this dialyzer? Basically, this artificial kidney, it is based on the principle of diffusion only. What is done from a convenient artery, right? The blood will be taken from a patient. It will be passed through this dialyzer, having the dialyzing fluid. And what will happen? See, in that dialyzing fluid, its concentration is just like plasma, right? Its concentration is just like plasma, except the nitrogenous waste. It is having everything except what? Except the nitrogenous waste, right? It doesn't contain the waste, otherwise it is similar to plasma, right, but without the waste product. So what is this dialyzer, beta? This dialyzer contains a semi-permeable membrane. I'll explain it here also, but I'm telling you the general principle. So basically, and blood pump, like basically we semi-permeable membrane. So basically this blood pump, basically you need a source of blood as well. So from the convenient artery, from that patient's any convenient artery, what we have to do? We have to take the blood. Now in that, because outside the body, blood will get what? Blood will get coagulated, blood will get clotted. So what will be added here? Heparin-like compound, right? In that blood, what will be added? Heparin-like compounds will be added, because heparin is what? Heparin is the natural anti-blood coagulant. So the compound like that will be added in that blood. And right? Okay. So when this blood will be passed, right, through this dialyzer, the dialyzer is having the semi-permeable membrane, and like here in the case of, in the NCERT also they have mentioned that cellophane tube is there, blood will be passed through these cellophane tube, so they are semi-permeable in nature. So this is what the dialyzer is. So in its surrounding, what will we see? We will see the dialyzing fluid. What are we going to see here in its surrounding, beta? We will see the dialyzing fluid. So as I said that this dialyzing fluid will have the concentration similar to plasma, right, except waste. So its composition is similar to plasma except the nitrogenous waste. So when this blood will pass through it, as per the concentration gradient, that nitrogenous waste will come out here in the dialyzing fluid. And after all that filtration, my dear students, right, after all that filtration with the help of a vein and after adding anti-heparin and by bringing the blood to the body temperature, it will again be passed into that patient's body. Is that clear? So that is how this artificial kidney works. So here it works on the principle of the diffusion. So blood will be taken out, blood passes through the dialyzer, right, which is having the, on the other side of that membrane there will be the flow of dialyzing fluid, right beta. So urea, creatinine, excess of salts, right beta, they will move here, right, and the clean blood will be returned. Is that clear? Clean blood will be returned. Is that clear? So this is what dialysis is. Right, this is what dialysis is. Dialysis is. Okay, dialysis needs dialysis. Right, this is what dialysis is. So dialyzer, dialysing fluid. So there is the blood pump. So this is what you can see here. So blood will be drained from the convenient artery, pumped into the dialyzing unit. Right, so this is dialysis. So dialysing unit is there after adding the anticoagulant like heparin. So unit will have coiled cellophane. So cellophane is semi-permeable. So it is surrounded by this dialysing fluid, right? So it is having the same composition just like plasma, but it doesn't have the nitrogenous waste. So this cellophane tube, the cellophane membrane beta, it is porous. It is having pores. So as per the concentration gradient, things will pass through it. So as the nitrogenous wastes are absent in the dialysing fluid, these substances freely move out. Is that clear? Is that clear? So that's what you can remember, right? So then cleared blood is pumped back to the body through a vein after adding anti-heparin. So baki renal failure of kidneys, they are not at all working. Transplantation is also the option, right, students? And what are renal calculi? So these are basically the kidney stones. Okay, so stone or insoluble mass. Again, from the disorders part, for sure you will get the question. Right, you will get the question. So stone or insoluble mass of crystallized salts like oxalates, etc., they are formed within the kidney. Again, it is because of the water, like the kind of water and diet, multiple things affect that. Right, beta? Multiple things affect here. So this is the renal calculi. Understood? Right? So this is also known as urolithiasis. Okay. Baki glomerulonephritis, that is the inflammation of glomeruli. It can be because of any pathogen also, right beta? So again, that is again a previous year question. So any infection and injury in the basement membrane can cause this. And if glomerulonephritis continues, it can also result in the kidney failure. So this is all about this chapter. Right now, I'll share the PDF in the Telegram group. Okay, beta, in our official Telegram group. And even if you will see that in the description box, they are having a link, right? You'll get the, yes. So there is one link for the Unacademy test series also. And other than that, you guys can see the practice question links are also given in the description box. So please click there, solve the questions, right? Solve the question. So, and make sure that you understand everything. Okay. Right, so I'll provide you the notes in our official Telegram group, that also you can join from the link given there. Okay, so this is all about this chapter. Next is morphology of flowering plants, because this is the chapter that you all are requesting too much. So that I will take tomorrow, and that too in a one shot, right? That too in a one shot. Okay, so tomorrow morphology of flowering plants, plus I will put the recording part of anatomy as well. So tomorrow in one shot we are going to finish it. Okay. Done. So take care. Bye-bye. This is all about this chapter. Keep studying and study well. Please practice the question. Just check the description box. There is a link for the